view it afterwards. And let's see. Okay. Well, thanks everybody for joining us. It's great to see all these folks with us here this morning. Appreciate you tuning in. And so this is the 2022 Ohio Hemlock Woolly Adelgid Task Force meeting. For those that don't know me, my name's Tom Macy. I'm the Forest Health Program Administrator for the ODNR Division of Forestry. And just a little bit of background on the group. So I was just looking back at my records, and we had the very first meeting of this group back in 2013. I didn't realize it was that long ago, but that was the first time this group got together. And kind of the purpose for this meeting was just to bring together all the partners and folks within the state that might have an interest in or manage hemlocks or have an interest in HWA or hemlock conservation throughout the state. So we can get together, at least on an annual basis, and just kind of share out how things are going, what we're finding, what's working, what's not. Just an opportunity to kind of report out on how things are going as far as hemlock pests and hemlock conservation is going through the state. So, the group currently, I think we're around 80 to 85 people and seems to be growing all the time. So that's great to see. Obviously, as HWA and other hemlock pest issues are moving into the state and becoming more of an issue, more and more folks are paying attention to it and working on managing them. So the last meeting for this group was back in 2019. We met in person at Mohican. This meeting has been held all around the state. I think we've had a couple here in Columbus. The last one was at Mohican. We've done one at Hocking Hills Lodge before it burned down. So hopefully we can get back together in person again. Maybe the next meeting we can do that. But we haven't met in a while because we had a little worldwide pandemic and some other things that got crazy and kept us from meeting. So I apologize for that. But ideally, going forward, we'll meet at least annually is my goal for this group. So real quickly, as far as working Microsoft Teams, if you're not familiar with it, at the top, there's a little chat bubble. If you click on that, you can type into the chat and feel free to share questions. If you wouldn't mind introducing yourself in there, your position, your organization, that'd be great because not everyone might not know everybody here. I am recording the meeting, so we'll make that available afterwards. I'll send a link out where we can access the recording of this meeting. When you're not talking, please mute your mic. So there's a microphone button up at the top that you can click to mute and unmute yourself. When you are talking, if possible, it'd be appreciated if you could turn your webcam on so folks can see you. And then finally, I should have the agenda up and being shared right now so folks can see that. It was included with your meeting invite, so hopefully you have that readily available. But I'll also drop the agenda into the chat right now so folks can access that. Well, once it lets me do that, I'll put it in the chat so you can access that. All right, and so I'll be moderating the meeting, and to keep things on time, once folks reach the end of their time slot, I'll jump in to let them know the time's up and we need to move along to the next talk. But be appreciated if you could just keep an eye on the clock, make sure you don't go over on your time. We will have a 20-minute block after all the updates for question and answer and discussion. So we'll hold all questions on the updates until that time, which starts at 10 after 10 this morning. But, so if folks have questions, make a note of those questions so you can bring that up during that session, or you can type it into the chat and the presenters can answer it in the chat in the meantime. And then for any speakers that are giving updates, if you would, please introduce yourself when you start your update, just so folks know who's doing the report out in addition to the agenda. But with that, I think we'll jump into the first presentation for today, which is us, myself and Michaela Height, doing an update from ODNR Division of Forestry. So I'm going to stop sharing my screen, and Michaela, I think, is going to share the presentation from her end.
Yep, pull that up now. All right, how does that look?
Looks good from here.
Awesome. I'll just introduce myself also. My name's Michaela Height, as Tom said, and I'm a forest health forester with the Division of Forestry, and I'm the crew leader implementing our hemlock work, and I'm stationed out of Hawking State Forest.
All right, so yeah, Michaela and I are going to kind of trade back and forth presenting these slides, but just to start off, wanted to share this statewide map for HWA distribution, just to update folks on the latest findings of HWA in the state. So there have been a few since our last meeting in 2019 to update on. So you can see on this map sort of the reddish orange and the red counties all kind of up in the northeastern part of the state are recent findings of HWA. So, you know, Mahoning County was discovered in 2020 at Mill Creek Park there. And then we had several counties in 2021 where HWA was first found. That'd be Stark, Columbiana, Jefferson, Portage, and Summit. Some of those were findings of HWA on yard trees on private land, and others were public land. So, for example, Columbiana County was a county park as well as a state nature preserve and state forest. And then Summit County was Sand Run Metro Park near Akron. So that's just an updated picture of where we know of HWA infestations currently in the state.
So just to give a little more info on the Columbiana County new HWA findings. So it was, there were some reports of HWA sent to us, and our hemlock crew went out to Columbiana County and met with Matthew Smith and Ryan Moss to survey the hemlock in 4 different public lands in that area. So we went to Beaver Creek State Park, Hellbender Bluff County Park, and Sheepskin Hollow State Nature Preserve, which also has some of Beaver Creek State Forest adjacent, and Beaver Kettle Farms, which is a conservation easement. And HWA was found at 3 of those 4 locations. We did not find any so far at Beaver Creek State Park. And so this became an opportunity for us to do kind of a collaborative hemlock treatment training. So in October last year, we did a treatment training with DNAP and Scenic Rivers in the county parks there and the Division of Forestry staff, so that we could instruct others that are more localized in that area on how to carry out hemlock treatments. I think we ended up treating about 50 trees that day, and I know Matthew Smith and Ryan Moss continued with some additional hemlock treatments in that area. So Hocking Hills, we have, as of the last winter, we had 17 known infestations on state lands in the Hocking Hills region. 8 of those are in Cantwell Cliffs area, and 9 are in kind of the lower Hocking Hills in the state forest and one in the state park, it's kind of south of Old Man's Cave. So this is kind of an overview map showing where going into 2021 treatment season where we had known HWA infestations through Hocking Hills. So that helped us kind of inform our treatment plans then for the year. Oh, there is a, in addition to those, through our survey efforts going on this winter, just in the last couple of weeks, there's a new infestation found just kind of on the edge of Conkle's Hollow and near our headquarters. So that's a new one for this area as well that we'll be looking at in our next treatment plans. So these couple of maps just show our those infestations from that overview map, but kind of zoomed in a little bit and showing our hemlock stand priority layers as well. So you can see those the blocks of yellow and orange and red. Those show the different priority of our hemlock stands in the area, and so that's based on the on recreational use, high quality streams, and presence of threatened or endangered species as well. So that's kind of how that was formulated. And this is when we went to plan our treatment areas for 2021, we were looking at where do we know our infestations are, and then using these different priority of the hemlock stands to determine how we're going to approach treatment. In 2021, we completed 7 different treatment units covering 20 acres. So these treatment areas were more focused in on where our infestations were. Rather than being a larger area, we did more intensive treatments, going treating all trees down to 2 inches in DBH, where we had known infestations. So that is in hopes to limit the spread from those known infestations. So we would treat all trees around the delineation of the infestation, and then about a 100-foot buffer around that. So we treated 2,174 trees in Hocking Hills. 2 of the treatment areas were on State Park in Cantwell Cliffs, and the rest were on State Forest. And we used our 3 treatment methods of soil drench and basal bark using the 2F imidacloprid formulation, and then a trunk injection method as well using a Zytect, which is another formulation of imidacloprid.
Okay, just want to quickly give an update on some of the progress we made with biological control releases for HWA recently. So looking at the Laricobius beetle species, that's Nigrinus and Osakensis, which mainly feed on the Sistens generation of HWA in the fall and winter. We released 1,500 Laricobius nigrinus in 2020 at Lake Catherine State Nature Preserve. And in 2021, we were fortunate to get quite a few, and we did releases at Lake Catherine. The first releases at Sand Run Metro Park in Summit County, and about 500 Laricobius osakensis also at a new site at Cantor's Cave 4-H Camp in Jackson County. There were some great opportunities for us to collect these biocontrols, both in Maryland, where there's pretty well-established populations of Laricobius nigrinus, as well as I was able to participate with the Forest Service on a collecting trip to Washington State, which is the native range of Laricobius nigrinus, and we're pretty successful in both locations at getting a good amount of beetles back to Ohio. So at this point, we don't have evidence of major establishment of these biocontrols just yet. We're continuing to monitor, and we're hopeful that given enough time and enough releases that these will get established in certain areas, but we're continuing to monitor that. In addition to the Laricobius beetles, this is kind of exciting news. For the first time, we were able to acquire silver flies, which are now in the genus Leucotoraxis. They were previously called Leucopus, but they were reallocated into a newly named genus of Leucotoraxis. These are native to the Pacific Northwest, and they seem to be a really important predator of HWA in that part of the country. And so these were made available to us through cooperation with the U.S. Forest Service. The hope is that they're going to be an important predator on that spring progrediens generation of HWA. And so we were able to do releases both at Sand Run Metro Park in Summit County, as well as Lake Catherine State Nature Preserve. So the goal here is to try to get the silver flies established along with Laricobius, so they can work together, and they sort of are feeding at different points and different generations of HWA to get those established to really make a difference in the HWA population. We also wanted to mention this fungal disease that we're starting to see pretty significantly in a few areas, mainly in southern Ohio, called Rosalinea needle blight. It's a really interesting fungus. You can see in that top left photo, it kind of forms this weird fungal mat on the needles. And then on the underside of the needle, sometimes you can see these dark spherical little fruiting bodies of the fungus. But it's been causing some pretty significant mortality and defoliation on the lower branches. So I would say, from the ground to about 20 feet up is where we tend to see this impacting the needles. It's definitely in some areas killing saplings and seedlings. The large established overstory trees aren't seriously impacted because their canopies are just fine, but it is hitting the lower branches. And it tends to be in those low-lying areas near water, those kind of low slopes near creeks or streams where we're seeing this the most. Not a whole lot available online about this one. There's a few fact sheets from North Carolina and Georgia. So it seems like with, lately things being, more wet during the growing season is what's leading to an increase in this fungal issue. So something to be on the lookout for. Just quick mention, we've moved to ArcGIS Collector with our field crews to do our survey and treatment data collection. So if folks are interested or have questions on that, we're happy to talk about that. Also a quick mention, I think Rachel Kapler may talk more about this later, but we did do an effort to collect some hemlock seed this year. And the idea is just to preserve some of that hemlock genetic material, and we sent that to the National Seed Lab. We created this little hemlock sieve with some wood and hardware cloth, and it was really effective at getting the seed separated from the cones. So happy to talk with folks more about that if they have questions.
So just to update on our survey progress this winter, we've done some thorough survey at Lake Catherine State Nature Preserve, at Shawnee and Mohican as well already this winter. At Lake Catherine, we did find that HWA has spread. It's been a handful of years, I think, since this was last more thoroughly surveyed. So we would consider Lake Catherine to be generally infested at this point, but there are some areas that are heavier than others. Overall, it's mostly a light infestation with some areas that are heavier in HWA. And at Shawnee, we found more HWA in some new areas, and in Gas Hollow in particular is an area where we may be seeing our first signs of decline from HWA. It's hard to determine that for sure with several factors there, but what we found there did appear like it might be our first kind of evidence of decline from HWA in the state. At Mohican, we have still not detected HWA so far. And Shade River State Forest was just surveyed recently. HWA is there. It has been found there previously. It's not new there. And Hocking State Forest, as I showed in that overview map, we're still in progress working on all of the Hocking Hills, hemlock. We have found some new infestations so far, though. So that's kind of where we're at right now, where we've finished most of our other areas and are focusing our efforts back into Hocking Hills for our survey. And then just kind of finishing up, our priorities moving forward are to continue with integrative pest management practices with incorporating our chemical treatments and biocontrol options and following our Eastern Hemlock Conservation Plan. We're going to continue planning our chemical treatments for our high priority hemlock stands where we have known infestations. So after we complete all of our survey efforts on the state lands, we'll be looking at the different priority hemlock stands to figure out, you know, where we need to focus our efforts first and how we're going to carry out those plans. And we also want to continue by offering more hemlock treatment training workshops. We know that as we continue to move forward in HWA spreads, we need as many partners like all of you as possible working on this together so that you know, we can work on this hemlock conservation more as a collective effort and get more people involved. So that's something that we're hoping to do more of over the next year. And then finally, just want to give a shout out to my hemlock crew that we've got at the Division of Forestry. So we had a team of 5 of us doing most of this treatment work through the summer, and that was 2 intermittent staff, Taylor Dawes and Addie Ramsey, a college intern, Lydia Dodson, and then myself and Roger Hendershot. So that concludes our presentation. Thanks.
Great. Thanks, Michaela. And again, we'll hold questions till the Q&A session at 10 after 10. So we're going to move along and go to Derek Cooper, who's going to talk about some of the work going on with the partners up in northeastern Ohio under a Great Lakes Restoration Initiative project. And I can see your slides there, Derek.
Excellent. Thank you. All right. Let's see if we can. Boom. All right.
Awesome.
All good? All right. Excellent. So hello, everyone. I'm Derek Cooper. I work with the Nature Conservancy, and I'm our Grand River Restoration Coordinator. I am the one that is overseeing our Central Lake Erie Basin Hemlock Protection Grant that is GLRI funded, which we will get into right now. So TNC was awarded a GLRI grant to protect hemlock in the Central Lake Erie Basin, of which includes everywhere from Lorain County as the western boundary all the way to the Pennsylvania border. So this is a collaborative project within the Lake Erie watershed. We have a mix of federal, state, local, and private partners, and it is GLRI funded. So the main purpose of this project was to develop a distribution of where our non-native pest species affecting hemlock were located and try to understand their densities, and then try to implement control methods based upon what we were finding in order to minimize the overall impacts to our forest health and high quality streams. So the summary of this project was our deliverables for this project were to survey 1,000 acres of hemlock forest for non-native pests, complete 600 acres of pesticide treatment, and perform 4 presentations. And mind you, this project was this is over a 2-year period, which actually got an extension. So now it goes until 2024. So we have quite a lengthy amount of time to complete all of our goals. So what has this project accomplished thus far? TNC and the partners across the region, which include some of our county park districts, other private nonprofits, we've been working together, and thus far we've actually been able to treat about 460 acres of hemlock for HWA and elongate hemlock scale in the region. Most of all of our treatments have been basal bark or injection. There's been 1 or 2 organizations that have, used soil drench application, but I know for my crew, our primary application method is basal bark. And to date, our collaborative efforts across the region, we've actually been able to survey 2,000 acres. Some of that is area that we have surveyed in previous years and then resurveyed, but a lot of it is new acreage because we're trying to get as much footprint out there as we can and really try to scour the area and find these pests. As you know, I don't know how many people are familiar with Northeast Ohio, but we don't have expansive hemlock forests. Most of our hemlock are really confined to a lot of our stream corridors, steep ravines, north-facing slopes, stuff like that. We do have 1 area of hemlock yellow birch swamp forest in Ashtabula County, which is a high priority area. So hemlock woolly adelgid in Northeast Ohio. So when this project started, we had 1 known location for hemlock woolly adelgid. Tom and Michaela showed a very nice map of the timelines for when those things were first found. So since the beginning of this project, we've actually added 4 new locations within the Lake Erie watershed. And then we also have added 2 locations in Mill Creek Metro Parks in Youngstown area. Mill Creek got added into this project later on because we, upon our first initial survey efforts, we weren't actually finding many pests. And so a TNC staff member was going on a casual hike in Mill Creek Metro Parks and happened upon to find some adelgid on a random branch and notified us. And we actually got approval from the Forest Service to expand our project beyond the bounds of the Lake Erie watershed to include Mill Creek Metro Parks. So we've actually been able to go down and help them out as well with some treatments, which has been really phenomenal. So this is just a map of where some of our adelgid populations have been found in the region. So you'll notice the 1 diamond with the red circle around it. And that's the initial population that was found in the region. So since then, you can see the locations down in the bottom right. That's the Mill Creek Metro Parks in Youngstown. And then everything else you see is in the Lake Erie watershed. The big in the kind of bottom left, that big blue area is the Cuyahoga Valley, which includes the National Park, Cleveland Metro Parks, and Sand Run Summit Metro Parks. So we have the very southern population of adelgid found there is in the Sand Run Summit Metro Parks. And on the very northern end was actually a private property fairly adjacent to the national park. So we have a lot of survey efforts we want to continue to conduct because this is, going to become a high priority concern. And then up in Lake County, you can see that 2 new populations were found. 1 next to the red circle is actually in the Grand River watershed, where the initial finding was actually in the Chagrin River watershed. So that's one of the new first recording of adelgid in that watershed, which is a great concern. There's a lot of big hemlock ravines in there. So we'll be hopefully performing some treatments this spring to start helping out with that newly found population. And then the one up on the lake is a very weird, isolated, planted hemlock kind of situation. And so Lake Metro Parks is going to go ahead and they're going to treat that this year as well. So elongate hemlock scale kind of has been the more established pest between the two in Northeast Ohio. When we started this project, Cleveland Metro Parks, Lake Metro Parks, and Holden Forest Gardens had already had known populations, and they had started to deal with them in some capacity or have been monitoring them for to evaluate how they're increasing in severity. So what we have found is that EHS has a slightly broader distribution. However, it does not appear to be as intense and widespread as we once feared. We were afraid that it might have been throughout the entire region, and instead we found that it actually was fairly reduced in distribution compared to what we thought. This map's a little confusing because I don't have all the data for locations in one place. So you can see that there's some green and purple squares. Those represent places that the Nature Conservancy had surveyed, found scale, and recorded them by intensity. So anything that's in that magenta kind of hue is actually a high density infestation of elongate hemlock scale, and the green is a low or mild infestation. And the red circle on the left is Cleveland Metro Parks North and South Chagrin. Those are two locations that we know that they're a scale. North Chagrin actually has some pretty intense infestations, which we've actually started to conduct some treatments for this in this previous year. So you'll see that there's kind of some random green dots scattered throughout. Those are just acre block units that we actually found 1 or 2 scale in. So it does seem to have distribution across the area. It's not rather intense, except in this area right up here where my cursor is. And then Lake Metro Park staff this year, also the upper right-hand red circle, discovered new populations of elongate hemlock scale traveling up the Grand River, moving north and east. So we are starting to see that the elongate hemlock scale seems to be moving rather quickly. Some of the Lake Metro Park staff have suggested that they've noticed that the intensity has increased over the last year. They've noticed a kind of a jump in the amount of scale they've seen. So we're going to be evaluating that over the next year and really trying to come up with a game plan for the region. So I just want to say thank you to all our partners as well. This project would not be happening if it wasn't for the amazing partnership that we have within our region. I can't even count the amount of people that are involved in some way, shape, or form, whether it's through on the ground work or whether it's through the partnership of the Lake Erie Allegheny partnership that we've utilized to gain some funding to use as match for our grant. This is a big regional effort, and it takes a lot of people, and we're really, really thankful that we have the wonderful partnerships that we do. And we also thank Tom for all the help that he's provided us up here in Northeast Ohio. So with that, that is all that I have to present on.
Cool. Thank you, Dave. Appreciate that. So getting a little ahead of time, which is totally fine. We can now move to Rob Curtis with Summit County Metro Parks and Sand Run in particular. I see you there, Rob. You have anything to share as far as your screen, or you good to go?
No, unfortunately,
okay.
I didn't put together anything formal.
No problem.
I apologize.
Oh, that's fine.
Yeah, my name's Rob Curtis with Summit Metro Parks. I'm the ecological supervisor for our conservation department. We are a bit of an anomaly as far as the group goes. So obviously interested in our hemlock, our eastern hemlock. But since emerald ash borer and sort of the ash EAB swept through, and we started treating with some of the neonics, treating some of our ash, and became skeptical of some of the collateral damage to native host species and other biodiversity around those areas. So as a park district, at least our conservation department anyway, we are fairly skeptical of pesticides in general, and done a good bit of research. I know neonics are fairly safe, but they can cause collateral damage, obviously, to other arthropods that might host on those species, as well as occasionally get into drainage systems. We have about 4 decent hemlock populations in our park district, as well as other scattered plants. And obviously those in Northeast Ohio, those seem to, well, they are in our park district associated with some of our higher quality terrestrial habitats, some of which we know have new species to science and very rare invertebrates. So that in combination has sort of caused us to shy away from using some of the pesticides that the rest of the group is typically involved with. So so we have a slightly different approach, and are to try to counter some of the host species issues with hemlock and some of the habitat that the hemlock creates. We decided to go with underplantings of Western hemlock, which is of course not native to Eastern U.S. It mimics in a lot of ways and is very similar to our Eastern hemlock, and is unlikely to do well here because it requires more moderate temperatures, wetter conditions. We did start this in our largest hemlock stand. We underplanted 200 Western hemlocks earlier this year. We have them all GPSed and are monitoring their progress. A lot of, probably about half of them so far have died. If they ever begin to cause problems, we could take them out, but we want to just sort of have them in the system, in the mix, while we may have Eastern hemlock decline, and we can sort of morph that habitat into another hemlock system. We have also toyed around with white pine and some other species that could sort of mimic that habitat, but Western hemlock obviously would be able to host a lot of our hemlock natives here as well, as far as invertebrates. So we did that. Then, you know, our other approach is just to let natural selection take place, rather than, you know, potentially treating plants that may not be well suited for natural selection and adaptation past the hemlock woolly adelgid, and keeping bad genes in the mix. We'd rather sort of let that take place on its own, let the fittest hemlocks continue to reproduce, and I know they do. I've been following some hemlock stands in central Pennsylvania for at least 3 decades, and there are plenty of hemlocks still in those stands. So we have time to change our minds. Anyway, that's our approach so far. And then finally, to use biocontrol, we do feel at the parks, regardless of whether it's plants or what invasive species we've been managing for decades, biocontrol is really the only realistic solution for invasive species control, especially at large scales. We just can't keep up with the continued input of invasive. So we're pretty happy that there was some biocontrol options, and that Division of Forestry was willing and ready to work with us right now. And so I'm going to back up just a little and say, kind of give you the status of HWA in our parks. Early this year, a volunteer found hemlock woolly adelgid in our largest stand in Sand Run, smack in the middle of Akron. TNC came in as part of their survey and found that infestation was pretty severe. Been around a while, probably 10 years or so. So we had a well-infested stand of hemlocks, and it was one of our largest, about a 25-acre stand. A lot of regeneration, very healthy hemlock stand in general. So it fit the bill pretty good for biocontrol release. There were a lot of low branches that we could recollect and resurvey from, and the trees still looked healthy even though they've been infested for a long time. So according to Tom, it seemed to fit what they were looking for biocontrol releases, and we were fortunate enough to be able to start that. And then, so this spring, we, in sort of 2 installments, we input about 600 silverflies. And in the fall, we were able to get 2 installments totaling about 2,500, a little over 2,500 Laricobius beetles. And the beetles, anyway, seemed to go to town as soon as they were released on, feeding on the adelgids. So we have high hopes that those will both take, and not only maybe be able to control some of the adelgid there, but be able to be a source for other sites, for others that might be interested in the area. And that's about all I had. I don't know if anybody has any questions. Okay, thank you.
All right, thanks, Rob. Appreciate that update. So moving along here, our next presentation will be from Joe Mosbrugger with Crane Hollow Nature Preserve. You ready for that, Joe? I think you're on mute. I can see your slides, but can't hear you yet. Mute button, it should be up at the top, the little microphone. Still not hearing you. Can you type into the chat? Can you give us a thumbs up or thumbs down whether you can see the microphone button, Joe, if you can hear me? We had him earlier. He was able to talk fine earlier. I'm not sure what's happening now.
His video looks frozen, Tom, so he might have lost connection.
Yeah, I think that may have happened. Well, maybe what we'll do is try to get, oh, here he's trying to get back in. Let's see if this works. We are a little ahead of schedule, so that's good.
Can you hear me again?
Yes, we can hear you and see you.
Sorry, I had to restart Teams, and I knew I was muted, but I couldn't do anything to stop it.
Gotcha.
So we'll try that again.
Okay.
Great stuff. Okay, I'll try and share, and hopefully things don't go bad. Can you see my slides?
Not yet.
Okay, let me What the hell happened here? Let's go to anything.
No, nothing yet.
Okay. I had actual teams running before. Now I've got. I just tried to do the web browser this time, and so I might not be able to share from there. So let me.
Okay.
Yeah, maybe if I apologize.
No problem. We're ahead of schedule.
Yeah. Let's crap. Okay.
I thought there was an option to share from the browser, but
yeah, I figured there would be. Oh, I think let's see if this works. We'll see what happens. Maybe somebody else can go. I can figure this out. Like I'll try and restart Teams.
Okay.
Because otherwise people are just going to get angry at me.
Yeah, no worries. We'll just bump ahead and we'll circle back and see if we can get you for the next job.
Good deal.
All right, cool. All right, Tim, I see you there. Are you ready to?
Yeah, I'm ready to go. Can you hear me?
Yep, can hear you and see you.
Okay, great. I'm going to try to share my screen here real quick. Let's see what I got. Okay. Okay, can you see my screen here?
Yep, I can see it.
See the report that I drew up then?
Yes, I can see you scrolling down.
Okay, great. I'm Tim Poehler. I'm one of the land managers here at the Arc of Appalachia Preserve System. We're a nonprofit land trust down here, and mostly in Southern Ohio, purchasing land throughout many counties here in Southern Ohio. Our headquarters is in Highland County. But we've recently even gone up above, Route 70 up to Holmes County. We just got started into the wetland complex up there at Killbuck Creek, so we're kind of all over. We've got a couple properties out in Hawking Hills. Here on this first map, you can kind of see, maybe the second one would be better. You can see this is a property in between Red Rock Hollow and Hamilton Hollow, mostly surrounded by state forest land there, nearby Camp Odeakwa and Old Man's Cave region. So it's about 40-acre block there, right in between there with the road run right through it. So we were able to get a little bit of funding from the Clean Ohio grant and got some money to do this treatment. This was my first time doing this, so I just wanted to give a big thanks to Tom and Michaela, and I think I see Stephanie Downs is on this call too. She was really helpful, helping me learn the ropes about how to go about all this. So I took this 40-acre block. We got it's about maybe a third to more has hemlocks on it of the 40 acres. And I was just kind of learning as I was going and figuring out what worked best for our situation. We just have a couple staff members, not a lot of resources, but just doing the best we can with what we've got. So what I did, you can see on this first map, we first did a survey, and I just split up the acreage by topography just to make it simple, so you can see those lines running through there. That's just kind of how the land split naturally. So me and a coworker, Ethan King, we went out there one day and basically just counted every tree we could find to get an idea of how many hemlocks are there, how many are there in the different size ranges. And that gave me a basis to try to start basing my treatment plan off of. So got about that, and that let me know about how much chemical to order and how much how to go about preparing for it. So we did that, split it up into these different areas, and then I decided to try to focus on, and after talking with Tom and Michaela and others about what they do, and because we're surrounded by the state forest, I was able, Tom and Michaela provided me with maps of where they had treated, and there were known infestations just in the state forest land on one side of it, and then fairly near on the other side, and we knew Camp OhioDNR had one. So we know there was nearby infestations. And while we didn't find any on our land directly, I felt it was prudent because we had this treatment funding, which was about to expire. We went ahead and did a treatment for all those trees that were 18 inches and up. In the upper, on this map I'm showing now, the upper left corner, that was more near one of the infestations on the state forest land. So we treated a few more, maybe down to 15 inches or so. But kind of did that and just traversed the property doing the treatment. Mostly soil drench. Did a few of the Cortex tablets though. So that worked out pretty nicely, and just did the same methods there using the, I think I attended one of Tom's, and I think Stephanie was there that day too, the learning how to do the treatments and just followed their protocols. So yeah, you can see there on the next map that we were able to use the Avenza app and mark where each treatment tree was, marked them with an orange dot. And then obviously we'll be monitoring that over the next 5 to 7 years to see how that's coming along. Let's see. I think that's kind of about it. Just a little overview there of what we did there. It worked. It went great. No problems. Again, with all the training received otherwise, and the answers to my questions that Tom and others had helped with, that really helped everything go smoothly. So that's for the Hocking Hills property. And then what's next for us is, I don't have a map of any of this, but my next focus is going to be Rock Run area. We have property down on the Rock Run watershed down near the Ohio River on the south side of Shawnee State Forest. So here I can stop sharing my screen. So we'll be doing a survey down at our Rock Run Preserve. We're probably going to do that survey sometime in February. So again, I'm still trying to debate on, it's a pretty rugged territory, so I got to work on the plans of how to get in there best, maybe splitting up teams, surveying different areas. But we'll just go through, try to look to see if we have any, identify if and where any known infestation sites are. And then I'll put together a treatment plan, and we'll probably go for treating at least those trees and maybe the ones around it. As I think Tom and Michaela were saying before, we know about ones in Shawnee, and I was not happy to hear that it sounds like there might be some decline down there in Shawnee. So we did have a volunteer last year who, I think it was last winter, he did find one of the hemlocks with it on our property. So we're going to try to, again, get that identified where the trees are and then put together a treatment plan. So anybody wants to come out and help with the survey, that information is on our website. I think we're doing it in later February. We're going to get out there and survey for these hemlocks or for the adelgids. So yeah, so we're just going to start there, survey, get a treatment plan together, and then go from there for the Shawnee properties. And then back to the Hocking Hills. Mostly that one that I spoke of is the one that we have the most hemlocks on, with surrounded infestations on other lands. So we'll keep our focus there. We do have a couple other properties in Hocking Hills, not major stands of hemlocks, but we're going to probably be doing some informal surveys on there just to keep it monitored. And if we see any need to take action, we'll do that in the coming years. And being part of this group has been very helpful to really get an idea of the larger regional effort. So that's been helpful. And before that's about all. And I just want to say, Rom, the previous speaker, I was happy to hear about your other methods you're trying. That's very interesting. I love to hear about how some of those other alternative options are going, and a continued effort to make sure to point out the collateral damage, because that was always the hard trade-off. When you treat these trees, you're knocking out all the other bugs and beneficial insects, native insects that are on or just around those trees. So I'm sure we all keep that in our mind, and that's the unfortunate side of this, but it's good to hear about lots of different options being tried out, and we'll see what works and what doesn't. So that's all. Thank you very much.
Great. Thank you so much, Tim. Appreciate it.
I can try and share again.
Yeah, you want to try again?
Yeah, if it blows up, though, I might just have to talk to you guys.
Okay.
I think that's one thing. I've got too many things open. All right, can you see my?
Yep. I can see the slides, and I can hear you.
All right. Thanks for being patient with me, folks. My name is Joe. I was lucky enough to grow up with hemlocks in my backyard in northeastern Ohio. Sadly, they do have a elongate scale on them, and every time I visit my parents, I check it out. And I never figured I'd get to spend so much time working with hemlocks in my life. Just a little background I wanted to give you on Crane Hollow here. I sort of like to say that we have some of the prettiest hemlock forest I think that's out there. So I feel really blessed to work here, but we have almost 2,000 acres of land we manage down here in the Hocking Hills. We are dedicated as a state nature preserve. Kind of where are we located? I kind of like to joke that we're right in the heart of the hemlock belt. If you look at the sort of shapefile Nicole created years and years ago, Crane Hollow, that little block of red, we are smack dab in the Hocking Hills. In some of the maps that Tom and Michaela showed earlier, there's kind of a U where it looks like there's no state land. That's where Crane Hollow is located. It's kind of interesting. They were talking about where the hemlocks are specifically found, like sort of up in northeastern Ohio in tiny ravines, north-facing slopes. They kind of behave differently in Crane Hollow. It's interesting. They really like the west-facing slopes, I feel like, in Crane Hollow and a lot of the Hocking Hills, but they really like our steep, sort of rocky, fun, beautiful areas, both above and below there. But we have hundreds of acres of hemlock forest to dominate, so it keeps us quite busy. And just, I will say, I think, to be honest, we've got some of the prettiest hemlock forest, sadly, eastern hemlock forest in the world. Sadly, that might change, and so we're trying to do everything we can to prevent it without harming the critters that live here. I think Rob made some good points. We are actively treating, but we do have some really just interesting both invertebrate species and then the vertebrates that prey on them. We get a lot of fun warbler species. We've got fun dragonflies that need these cool, clear streams. We get all of our wonderful little hemlock specialists. It's sort of interesting if you look at all these bird pictures that were taken by John Watts. I think every single one of these is actually found on a hemlock tree in the pictures. It's just sort of interesting there.
Hey, Joe.
Yo.
Your slides are not advancing for, I don't know if anybody is seeing them advance or not, but several of us can.
That's, What slide are we on right now?
Still your opening slide.
Oh my goodness. Because
you never went into presentation mode. It didn't look like
that's
on
a
my
shame.
end.
I'm on presentation mode on my.
Oh, maybe it's the wrong screen. Did the presentation come up on a different screen or?
No, I'm only using one monitor here.
Okay.
Let me. Son of a gun.
I can see your cursor now.
That's so bizarre. Oh, hang on a second. Let me. Let me stop presenting and try again. I don't know what else to do. Yeah, I'm kind of let me.
Joe, this is Rick. There's two different ways you can share your screen. There's one that's your just general all open windows, or you can choose a just a specific one. So, in
Yeah.
teams.
Yeah, that's what I did. I shared a specific. So let me maybe
Okay.
try the other way. That's a good point. Boy. Okay. Okay, I'm opening the share tray again, and then I'm clicking on the. Let me go back to. All right. Can you see this right here?
Yes.
Can you see my screen?
Yep.
Okay, are you seeing slides this way? Like,
Yes.
I'll just do it this way, and it's not going to be full screen, but I don't know what else to do because it's
That works.
it's all
The other
screwed up.
thing, one other thing you could do is down at the bottom right where it says 60%, there's the button next to
Yeah.
it. That'll make the picture bigger, and we can actually, some of it's a little bit harder to read not the
Yeah.
60-80%, but that little button that makes the presentation cover the whole screen. It's the one to the left of the 74%. That little tab there.
Oh, now I zoomed way up to where I was.
Okay.
Yeah. And you guys aren't seeing these slides though.
We still see it, but not in presentation mode.
Okay, but you're not. But I'm going through slides and okay. I don't know what.
No, not seeing it.
All right,
now,
so we'll just try it this way. I don't know what the
Okay.
deal is, but
That's cool.
blame the Hocking County internet. I don't know what to say. But yeah, you missed some beautiful pictures. But I'll just try and move on. This is the map that I'm glad you guys spoke up though. But you can see where Crane Hollow is located there. There's some more pictures just with our ice formations. We do really have some nice forest there. And then here's our fun animal pictures. Again, the state species of concern, tiger spiketail, along with our Canada warbler. We get others like the black-throated green, blue-headed vireo, and the hermit thrush. It's kind of interesting if you look at the breeding bird atlas, probably the blue-headed vireo follows our hemlock forests more than any other bird, which is just kind of cool to see. Rob Curtis gave a talk just a little while ago. It's also sort of interesting. We actually have a federally threatened plant species here at Crane Hollow, and probably the hemlocks might sort of play a part in protecting this plant as well. We think that our hemlocks like a little bit of sun, but they need it cool as well. So just the fact that we've got these hemlock gorges that, again, trying to keep things a little cooler all in all, it's sort of fun that we have things like that. Moving on towards sort of what we're doing with HWA specifically, we've really looked for it really hard since I think back in 2014. We've used staff. We have really employed loads and loads and loads of volunteers. And the one thing I've sort of found over years when I'm trying to train people is we've really tried to show them what an actual specimen looks like. So I actually will I'll get a specimen from somewhere. Sadly, I can just get it from our nature preserve now. But the way we find hemlock woolly adelgid in Crane Hollow, it doesn't look like it does in the pictures. We're finding just like a single little dot of white. And so I really want to sort of train my volunteers. It's like, oh, that's what we're looking for here. I really strongly encourage my volunteers to use Avenza, or some of them will use other things like TopoMaps Plus or Gaia or just whatever, All Trails. As long as they can get a GPS track, it's pretty useful. But we try and split into groups, check specific areas. Then if we find it, we'll take waypoints, we'll flag it. And then we come back here and we'll have a good old time. We like to volunteer retention is sort of a big thing. So we try and bribe people with some snacks. And again, because we're a NGO, we can even give them some beer, either that I brew or what I find on the side of the road. So that's kind of fun. If you look at, I skipped ahead one too many, I think. Yep. But the other thing too, it's just a really good excuse to get people out in the woods. It is a beautiful place where we're able to work, and we find some really just beautiful ice formations. I've got 2 groups coming this weekend. I hope to show off fun things with them. But the other thing, just sort of doing this, we've found just loads of bats that like to hang out in Crane Hollow. Usually we see them in the rock crevices, but it's kind of fun. We actually found a red bat when we were looking back in December here, hanging out. It was really, really warm back then, so kind of crazy. But We'll also see salamanders in the springtime and ephemeral flowers. So it's just a good excuse to get people out in the woods. In 2021, we actually monitored 416 acres with volunteers. We actually did 15 days where we got help from some sort of outside source. Sometimes it was school groups, sometimes it was hiking groups, sometimes it was just my good old open sort of whoever wants to come comes. And last year, and I'll sort of say back in 2018 is where we first found it. I'll use my cursor down here in Silex Hollow. Not surprising because it was found also on state forest land just to the northeast there. Again, we knew it was sort of coming around because it was also in Spruce Run on Hocking State Forest land. So it wasn't surprising when we found it here, but maybe partially cold winters. We found nothing in then the following 2018, 2019, 2020, we did really well. But then in 2021, we really, unfortunately, had 3 new infestations. A volunteer found it on a single tree in Snyder Hollow, and then we found a pretty large infestation there in 2021. Moving onwards, what we've done so far this year, you can see in the black is what we've looked at just this year already, but now we've got some extra pink dots we found here. Again, we found it right up here in the very north. Not surprising, we found it close where we found it before, but we're seeing other novel things. And then Tom and Michaela and Roger, I'm sort of using this information just to let you know, I found it pretty much right on the border of state forest. I might have volunteers sort of go out there on our property and see how bad it is. This I unfortunately found just on a leisurely stroll where I wanted to look at ice and check things out. So it's getting worse, unfortunately. Our treatment strategy, we're not preemptively treating trees. What we're trying to do is just really focus on the infested area. Something we're doing a little different than a lot of other groups is we're using a lot of the Cortec tablets. We kind of like these just because they're light, and we also get a higher per acre limit with them. So we've been using those a lot. In our worst infestations, though, we've done a lot of stem injections just so the pesticide gets directly in the tree, and we're not, again, we're just not harming arthropods as much. There's the goal. You can see what we did in 2021. We probably just did a little under 300 trees. You can see by far where we did most of our treatment was in that Punch Bowl area where we had our biggest infestation. In 2022, we know that we're going to be doing a lot more treatment here. I'm going to try, and I've just sort of in questions, I've always been kind of a little frustrated with just the uptake of some of the stem injections. So I just wanted to get some advice from other people who have done this. But I think if we do it a little earlier in the year, it'll help us. I think especially on like sort of south-facing slopes. I think the soil was too dry just to get much uptake, and even by June, July, and on and on. And just finally to sort of say thank you to a whole bunch of people, various universities, the Ohio Certified Volunteer Naturalist. There's a hiking group called Feet on Fire, thanking folks at ODNR like Tom and Stephanie, who I've bugged for help before. Levi Miller with Bach Hollow, he came out on our first treatment day. But we've also just tried to really spread the hemlock gospel a whole bunch to a lot of different parties. So thank you for listening. I apologize for all the technical difficulties there, but thanks for being patient with me, everybody.
Thanks, Joe. Thought it worked out just fine.
Yeah, I'm sorry though. So I think I need to get a new computer.
No worries, no worries. You're good. Appreciate it. That was great. Okay, up next, we've got a 5-minute update from Dave Adkins at ODA. You ready for that, Dave?
Yes, sir. Okay,
cool.
Let me share the screen here. Can everyone see that screen?
Not seeing anything yet.
Okay, let's see here. Come on now. Oh, we're just having all sorts of fun today, aren't we? Has that come up yet?
No, nothing yet.
Okay. Let's see here.
Now it's coming up. Yep, I can see it now.
See it now? Okay, very
Yep.
good. I'm Dave Atkins. I'm Ag Inspection Manager, Plant Pest Control Section here in the Ohio Department of Agriculture. My main work has been with invasive species and so forth. I've worked with hemlock woolly adelgea with Tom for a long time, since that 2013 era started. I've also worked with gypsy moth and now on spotted lanternfly and several others. Right now, what I'm showing you is a map of the current quarantine counties here in the state of Ohio that we have listed for hemlock woolly adelgea. What we do is regulate the movement of nursery stock and so forth that may have adelgea on it. What I want to bring up at this point is that a lot of counties are having their spring tree sales, sapling sales. Please make sure you're purchasing your tree stock from reputable nurseries outside of the state or wherever you're buying them from. We did have a situation down in Hocking County here a few years ago that the hemlocks came in full of adelgea. So we had to do a lot of destroying on those. But currently, this is where we're at. We regulate the nurseries in these counties and so forth for the movement. If there is a positive that you feel for hemlock woolly adelgea that is not on this list of counties, please notify us. We need the coordinates of where it was found. We need a sample of the adelgea so we can verify it with our entomologists here at the department, and then we'll move forward with what quarantine regulations may need to take effect in that county. So that's basically where we're at with the Department of Agriculture and the quarantines for adelgea. So that's all I've got, Tom.
All right, thank you, Dave. Appreciate that. Let's see. So next we have Chad Fitton with Wayne National Forest. You with us, Chad? If Dave, you want to stop sharing?
I have.
There you go.
Okay.
Good morning, everyone.
Hey, Chad.
So, yeah, go through this here. Good morning. I'm Chad Fitton. I'm a forest silviculturist on the Wayne National Forest. Today I'm going to talk about the status of hemlock on the Wayne. Most of my talk is really around inventory that we need to do to get a handle on the situation. We have about 210 estimated acres of hemlock, and that's based upon the Nicole Stump mapping exercise from our thesis. A lot of those areas we need to field verify. We're finding that it's very representative in some cases and not in others, where it's picking up white pine or other. So we feel like we need to get a real good handle on the inventory process and update our stands layer associated with hemlock, because the two right now just aren't in sync. And so over the next 5 years, we're looking, as I mentioned, do a system of inventories. I've been collaborating with Tom Macy, and we're looking to utilize the same protocols as the state is using. And that includes the art collector protocols as well for monitoring HWA infestations, as well as if we get around to treatments. So in the past, we have been monitoring hemlock. It's not been a real formalized fashion, but we have years above here that where we have been going out. Mostly that's been in our Marietta unit. Just due to staffing and expertise and that type of thing. During that time, we were not putting a concentrated effort on it, and that ship is now turned. So it's in the process of turning, I should say. So distribution-wise, I know these maps are pretty poor. They don't really give a scale. We have 3 distinct units of the Wayne National Forest. Not much hemlock in our Athens unit. We have a couple small areas around New Straitsville, Ohio, around Perry County there, Hawking Perry. And most of our hemlock sits on our Marietta unit and our Ironton unit. So in Marietta, we have about 69 acres of mapped areas, and we've been going through confirming those and starting to do detection work, further work. In January of last year, so about a year ago to the day, we were out surveying stands, and we confirmed it on our Marietta unit as well as our Ironton unit the next week. And where we confirmed it is right around the area of town of New Matamoros, Ohio, which is about a half-hour northeast of Marietta on Route 7 on the Ohio River, and some remote areas there near the Ohio River. So again, we're trying to just do more survey work and confirmations of additional areas. On the Ironton unit, we also detected HWA in 2021. We have about 138 acres of hemlock scattered around the greater Ironton unit. The green federal land on this Wayne National Forest represents about 104,000 acres. And you can see the distribution of hemlock mapped areas is pretty widespread, about 46 areas. And so, again, we're in the process of just, trying to delineate which ones are actually hemlock and then do the associated survey and inventory work with those. We did confirm it around our Lake Vesuvius area, which is in this southern part of the district, about 7 miles north of the town of Ironton. Overall, our hemlock health is still real high. We think the infestations are relatively new. Infestation levels are light. Some considerations that we as a staff and team are going through. We've talked a lot about inventory. We are in early discussions about initiating NEPA for HWA treatments, and that gives us the environmental clearance to seek approval for those HWA treatments. We also want to, as I mentioned, initiate some biannual monitoring where we would visit the stands on a biannual basis on a rotation there, where we would look for and detect the presence of HWA on a biannual basis and track that over time. And then when we're ready, we would consider funding sources like U.S. Forest Service state and private funding or other funding to treat those known infested sites. And so it's a little early, but the good thing is we have it right now detected in just 2 locations. Doesn't mean that's, of course, where it is isolated to, but we feel like we're not too far behind the curve, and so we can work that into our considerations. That's all I have at this moment.
All right, great. Thanks a lot, Chad. Appreciate that. Okay, up next, we've got John Apt with an update on Worcester Memorial Park.
Hi, can you see me?
Yep, can see you and hear you.
Can you hear me?
Yep.
Okay, I do not have a presentation. on your screen, but I'd like to talk about our park. Worcester Memorial Park is a city park for the city of Worcester. Worcester is located about an hour southwest of Akron, hour and a half northeast of Columbus. The park itself is not in town. It's located a couple miles west of town on a little bit north of State Route 250. So I'm a member of the Friends Group. Even though this is a city park, the Friends Group takes care of the park to a great extent. We manage the trails, we organize hikes, we try to take care of invasives, and we're very involved in land acquisitions. So the Friends Group has about 250 members. We have a website that talks about the history of the park itself, and we have a map of the park on the website. So the park is about 422 acres, 350 of which are wooded. We have about 12 miles of trails. The core of the park is Rathburn Run, which is a relatively small creek, but with a pretty deep ravine, which drains into the Little Killbuck Creek. The Little Killbuck drains into the Killbuck, which runs south into southern Wayne County and Holmes County. So besides the ravine of the Rathburn Run, we have a lot of uplands. We've planted about 10 acres of prairie. We've replanted about 10 acres of agricultural field with trees. And we have an old agricultural field that we're trying to manage in some way. So the hemlocks are mainly found in the ravine on both the north and south edges of it. Plus we have some scattered hemlocks in the uplands, just isolated trees and small groups of trees. All told, about 5% of the park is in hemlocks, or about 22 acres. And in those 22 acres, we have about 10 stands where we've delimited the hemlocks, and these stands range in size from about 1.5 acres to 4 acres. So over the last number of years, being aware of the adelgid, we've done some very informal surveys just looking for it. It wasn't until last year that we did more of a formal survey, and we looked at about 18 of the acres. And in these 10 stands that I mentioned, we probably looked at from 1/4 to 3/4 of the trees in each stand. And at this point, we have not found any adelgid. So at this point, we're just going to keep doing surveying. We haven't done any surveys this year, but I'm hoping probably not until March, we'll do some surveys and try to get volunteers and teach people about the adelgid and see if we can find it. So anyways, that's all I have. Thank you.
Awesome. Thanks a lot, John. I appreciate that. Yeah,
Okay.
and for folks that may not be familiar with Worcester Memorial Park, if you're ever in the area, I'd recommend checking out. It's a pretty cool place. So, thank you, John.
You're welcome.
All right, so up next, we've got another quick update from Paige Weckbacher with the National Wild Turkey Federation. Are you with us, Paige?
Hi. Yep, I'm here. It's a little
Hey,
slow
Paige.
going.
You're good.
Hi, so I'm Paige Weckbacher. Like Tom mentioned, I work for the National Wild Turkey Federation, and I have a similar agreement with NRCS that service foresters do with Division of Forestry. So my job mostly is to promote NRCS programs like EQIP and CSP. So that's my main focus, but I also worked for the Division of Forestry for about 4 years, working specifically with hemlock stuff and treating trees in the Hocking Hills with all those folks. So I do have a good background with HWA and all that good stuff. So one way that I can kind of help with this mission, I guess, is state and federal agencies can't really check out private landowner properties, that kind of thing. So what I can do is kind of be a first responder If somebody has a question or wants me to check out a possible infestation on their property, I cover 8 different counties within Southeast Ohio, including Monroe, Belmont, Noble, Guernsey, Muskingum, Coshocton, Harrison, Tusk, and can also help in Washington and Morgan counties. So if y'all have anybody come to you and have, pictures of HWA, maybe need to know, the best way to treat, what chemical to get, dosage rates, that kind of thing, I can come out and check it out and kind of guide them from there. I can't actually do the treatments, but I can, get with them to positively identify what they have. And then if they want to order the chemical, I can show them how to mix it. I can, demonstrate how you would actually treat the tree, and then just go on from there and let them do the rest themselves. So as an example, I had a landowner in Newport, Ohio, which is pretty similar to, or pretty close to where New Matamoros is, that Chad was talking about. They had HWA in their, they kind of had a small forest within their yard. And so I went down there and helped them identify it, helped them figure out a game plan, and showed them, how to treat it. And within a day, I just kind of helped out and got that taken care of. So if y'all have anybody come and it's not necessarily in your jurisdiction to go to their property and check it out, I can definitely be a resource. So I just wanted to let you guys know that, and let me know if you have any questions.
Awesome. Thanks so much, Paige. Appreciate it. Yeah, that's obviously a really important piece to this, the private land side of it, because we've struggled with how to address that. And obviously, within Ohio, the vast majority of our forests are privately owned. So that is really important in trying to help out those private landowners when it comes to HWA identification and steps they can take to manage it on their land. So thank you for that. And now let's see, we have Andrew Boos with Columbus and Franklin County Metro Parks, Clear Creek in particular. Andrew, you ready for that?
Yes, I am.
Sounds good.
All right, I do not have a presentation either. I am Andrew Boos, ecologist with the Franklin County Metro Parks, and we have Clear Creek Metro Park, which is 5,300 acres just southeast of Columbus, about 45 minutes. 5,252 acres of that is state nature preserve, and there's probably around 1,000 acres of hemlock scattered amongst that, mostly down along the Clear Creek Valley and up through the ravines. And we do also have the species like Joe was talking about with the warblers and tiger spiketails. So those are major concerns of ours with the loss of hemlock. We started monitoring in 2014, and Katie Bennett, our park naturalist at Clear Creek, gets the kudos. She has headed this up since she's been there and has done a great job with our volunteers. And we started monitoring in December, so starts real early. Have 6 to 7 volunteers per outing, and they come out once a week, spend about 4 hours, which is really nice. Their goal is to visit all the stands every 2 years. With 5,000 acres, this is quite an undertaking. Katie is also starting a public program just to bring people in to initiate them into the hemlock woolly adelgid. And that one, she has the first one in February, and we're hoping maybe we get some more people interested in volunteering. We also have staff members, myself included, and other resource management staff that goes down and monitors on occasion, but the volunteers definitely, and Katie, get the do most of the work for us. Mapping is done through Avenza, and unfortunately I was hoping to have this being a little bit shorter, but as of Sunday we had our first detection of HWA at Clear Creek by our volunteer group. And so that kind of changes things. We are now not just monitoring. We will now move into what we're going to do for suppression. We will follow the protocols and be very interested in what everybody else is doing. So we'll be more in contact with the folks that are doing the biocontrols, pesticide controls. We're not new to this with the Emerald Ash Borer, so that's good. This isn't something we're just stepping into blind. And we've had several people go to the HWA workshop. So I think we're in pretty good shape with that. And I think that is about it. It's a very short, just a short presentation from Clear Creek. And we'll be in touch with everybody as we move forward to join the group of people who have had the infestations. Thank you.
Thanks, Andrew. Bummer to hear that about that finding at Clear Creek, but yeah, happy to help however we can if we can assist at all. So, all right. So our final update of this morning is going to be Rachel Kapler with the Great Lakes Basin Forest Health Collaborative. So Rachel, feel free to take it away.
Hey, excuse me, getting over a cold. So if my screen sharing is working properly, we have a summary here, which I'm excited to share. That's from last fall's collections efforts, like Tom was talking about, of hemlock cones and cleaning the seeds out and being able to send samples to the National Seed Lab. And to give you background on what I do, the Great Lakes Basin Forest Health Collaborative is new, sponsored by the Great Lakes Restoration Initiative. And I'm the coordinator to help make sure that our main focus, which is to help set up tree breeding programs for trees that need to be bred for resistance to pests and diseases, are able to be networked together and get going, which we are just starting to get things organized for hemlock. And so, of course, the first initiative there is to collect and have samples of the populations that are already there. And in Ohio, you can see that we have a good number of different locations, 21 different trees sampled in all. We have some Hocking County areas, Summit County, Monroe County, and of course, I did a lot of work over in Geauga and Lake County at Holden Arboretum locations. The Cleveland Metro Parks told me that they were able to collect from a lot of their locations, so Cuyahoga County is covered. I still don't have confirmation on how much extra they have so that we can send it to the seed lab. A couple of different preserves from the Cleveland Museum of Natural History. They let me go out to locations, so we got Ashtabula County and a couple of small preserves out there. Had some help from Lorain County Metro Parks, and so they were able to get locations along the rivers over there, which is great. And so altogether, we have 21 new tree samples taken from Ohio, and that's really great. What I really would like to try and focus on next season is to look at the counties in Ohio that have hemlock and see if we can make sure to try and cover at least getting some samples from each of the counties. So on a larger scale of all of Ohio, we have a good collection bank of hemlock seeds. And I also, of course, reached out to other states in the Great Lakes area, and a lot of hemlock talk is always about New Jersey as well. So we're in contact with them and got some samples sent from Catskill Mountains in New York, the New Jersey mountains areas where they have the hemlocks that are seeming to be more resistant to HWA. And Pennsylvania also has a lot of locations where they think their trees are more resistant. And so those samples have been sent in. And Wisconsin as well has their different pockets of hemlock. And so we're really glad that they were able to collect from them again. They've been doing that previously. And all of this just to let you know, since we have with the Forest Health Collaborative, me helping out different research and networkings to get together and help out with things. Some of this work will start with hemlock breeding research, some at Holden with Dr. David Burke, and that's in collaboration with the U.S. Forest Service and New Jersey. And so we're at that location helping out with making sure we can get some propagated here in Ohio and seeing how we can come up with proper testing to make sure just how resistant individuals are when we get those that we suspect are from the natural areas into a controlled environment. And so I'm just going to stop sharing here. And if you have any, well, if I can get back to my other screen here, there we go. So we'll be able to help out with any information that you guys have on needing someone to come down and show you how to do seed collections, or are interested in having a certain tree checked out because you think it might be partially resistant. I'm going to go into the chat here and enter in my email. So I know a lot of you have, heard of me before. I've been around last year making all the HWA meetings, but I know we have a lot of people here that I might not have met before. So go ahead and feel free to send me any questions. And also, I might be reaching out to some of you this year to let you know that I'm interested in making sure we collect some seeds from your neighborhood. And I'll be happy to answer any questions during our break that you might have on the Forest Health Collaborative, and if you want to be a part of it and things like that.
Great. Thanks very much, Rachel. Appreciate it. Mm-hmm. And that, I mean, I think that's a really important area of work, trying to identify potentially resistant or tolerant hemlocks out on the landscape that, could potentially be propagated in a breeding program and then used for restoration purposes. But obviously, in most of Ohio currently, the hemlock health, I think, is pretty good. So as far as I know, for doing that identification of those, lingering or resistant trees, probably would need to happen, to our east and south where there is more decline and mortality happening. But yeah, obviously the seed collections and keeping an eye out, where we have HWA, what the hemlock health is looking like is really important.
Right. Sorry, I get really excited about the whole process of the tree breeding program. And just to clarify, we're right at the very beginning stages here in Ohio. We're helping out other states that have it more along in their system so that we can find those individuals. And we're also thinking about making sure, we're able to know who's monitoring where in Ohio so that we're all, clear on where we could look later on when we end up having a little bit of issues with HWA. Usually it's best to find those ones that seem more resistant in a field of trees that are just totally infested. So sorry about getting everybody a little excited about that. So we're not in the position yet with hemlock to provide any samples to people, but what we're doing is making sure that everybody knows we're trying to get people aware that we're trying to initiate a breeding program and research that goes along with that, just like was done with ash trees, so that we can take a long-term look at keeping this specific Ohio population of hemlocks preserved for the long term. And so any that you think are resistant right now, it's just one of those things where we're going to have to monitor it probably for a while. But it's good to keep track of.
Yep, and I think that, did you see Tim's question there? I think you just addressed that.
Yeah, I saw that there, and I was like, oh, right, I forgot. Everybody gets really excited, and I have nothing, I'm sorry, to hand out right now. But yeah, we're even still trying to get to the stage where maybe we can do something with ash, but ash isn't even ready yet. So the trees just take a long time. They have their own time scale.
Yep. And there are, I'll say I'm aware of, like Rachel had mentioned, there's an area in New Jersey that they've been calling the bulletproof stand for years now that appear to have pretty good resistance to HWA. They've lost most or all of the hemlocks around that area, but these trees seem to still be doing well. So, you know, there's work starting to look at that stand and potential other resistant stands across different states and different areas, you know, to our east. So
that's part of the Holden research that I'm very excited. We got successfully a packet of New Jersey seedlings that they were growing over there to have Dave start his research on. And one other thing, I saw a comment about cone collection and hemlocks, and I know it's really, after my first year of experience this in person, it can be very difficult. And so especially a tree that's getting good light, yes, is the kind of tree that is good to collect from. And it is unfortunately found, I think, from the hemlock researchers in North Carolina, saying that the higher up the tree you can get the seeds, the better the viability is. And that can be very tricky. And so we'll have to come together and see what we can we can do about people who are potential tree climbers and may be able to visit to take out some branches down, or using some rope saws or something to get a branch to get after those seed bunches.
And I see the question there from Connie about inquiring about getting any seed from that New Jersey stand. I think they're kind of currently in the research phase there. There was a researcher at I think University of Rhode Island that or maybe it was University of Massachusetts, in any event, that they have propagated some seedlings, and they've planted them out experimentally in some different states to kind of monitor their performance over time. So, you know, we were not part of that study, but they've been planted out, you know, around the range of eastern hemlock to see how they do. So we're still just kind of watching that at this time.
Yes, we heard from them this last fall when I sent out requests for seeds to be sent to the National Seed Lab, and of course they've done that with their seeds. And I might the research that Holden, the Forest Service, and the state of New Jersey are in cahoots with might indicate that we'll be able to get some in Ohio to see if propagating seeds from there in Ohio works well, and then doing, of course, more research on that.
Excellent. So on the agenda now, we're in our kind of Q&A open discussion section till 10:30. So I don't think there's any outstanding questions in the chat that I could see. But if folks have questions from any of the updates so far this morning for any speakers, or just general questions for the group, you know, type that into the chat. You can unmute yourself and put your camera on and ask the group however you'd like to do that. So this is our time to open the floor up. I do see a question there from Connie. Do you have an ID or fact sheet for Rosalinea needle blight? Yes, there are a couple online resources, articles. They're pretty brief, and there's not a ton of information on that disease, but I'll put them in the chat here in just a second.
Tom, I just wanted to touch on the resistant stand in New Jersey, and that resistance is shown to be specifically for adelgid, right? There's not like a co-infestation of elongated hemlock scale in those stands as well?
Yeah, as far as I know, it's just adelgid. That the trees, you know, are infested with adelgid, but they don't seem to experience decline And I'm not sure about the status of elongate hemlock scale on those particular trees. I don't think they've seen them on there, but I don't know for sure.
Yeah, as far as I know, we're just dealing with HWA at the New Jersey stand. And we have, hopefully down the line, you know, it's going to be a couple years once we try to figure out how we can test for resistance with hemlock seedlings. With HWA, we might also be able to, at the same time, test to see if they can go against EHS as well, since we do have it near Holden, so they're readily available to be transplanted onto some test subjects. And yeah, there's some hope there that there might be the same mechanisms that fight against one will fight against the other. I had a quick question on the people who were using Avenza with their volunteers. Is that something where each location has made their own like project that people join in to log their data? Isn't that how Avenza works? Or is it, oh, that's ANIC data. Got the A's mixed up. So, but with Avenza, that's where you would have them download their own, a map that you provide them, and then they keep track of where they've been?
Yeah, I do this a lot. Can you hear me there?
Yep, I hear you.
Yeah, so what we've been doing at Crane Hollow is a lot of times I'll make a base map that shows kind of, sometimes I'll really try and zoom it into a specific area, and then I'll either text or email all my participants that map for that area, and then they'll email it back to me, and that's how we do it at Crane Hollow.
Okay.
Also, if they've got like an old school GPS or they use Gaia or something, they can still get me a GPX file. They just don't have like more sort of detailed, focused map that I can provide.
Right.
Yeah, Avenza is pretty convenient in that it is a free app. Anyone can download it. You know, if you have ArcGIS or, you know, some GIS program, you can create your own base maps for use in Avenza. Or I think there are places online, you know, where you could potentially purchase base maps for use in Avenza. But yeah, you know, it can be used by anyone. They can create their own data points on the map. They can draw polygons on there and then export it and email it to anyone after the fact. So.
Okay, great. Just wanted to check on that. I'm always hearing about who's using what apps and things, and so I'm trying to keep track of who's got what.
Yep.
So I had a question for Tim. Can you hear me?
I can hear you. Are you still here, Tim?
Yeah, go ahead. Sorry, I walked away from my computer for a second.
I wasn't sure if I understood this right. In Red Rock Bluff, you were treating trees that were not infested?
Yes, at Red Rock Bluffs, we were doing a preemptive treatment. As I was learning about the process, I was unclear about, do you do a survey? Do you do treatment? How do you choose what to do? And as I was learning about my area, as we did our survey, and then got familiar with what Tom and others had been doing in the state forests directly surrounding it, knowing that there was known infestations very nearby. That's why we chose to go ahead and do a preemptive treatment, and did a few more, went down in the DVH size of what we were treating that was closest to those infestation sites. And then did 18 inches and up DVH for the rest of the property. We were also had a grant funding that expired, and so we wanted to take advantage of that. So I think in other places, I'm not planning on doing preemptive treatments per se, but focusing on the surveys, information from other folks like you who have property nearby ours to inform, but probably going to focus on surveys. If and when we find infestations, then we'll treat those direct ones. But probably not taking a preemptive strategy in general. But it's an evolving process. So, but like I said, next we'll be going down to Shawnee, and we do know there's infestations down there. So I do expect to be doing a treatment at the Rock Run watershed potentially later this year.
So how close were those infestations to the areas that you treated? A few miles or?
Yes, directly adjacent to our property, certainly well within a mile, maybe even a quarter mile from our boundary lines. There was one in one corner, known infestation just on the other side of the boundary line. And then in the opposite corner of the property, there was a known infestation on the state forest, a little further off than the other corner, but still very close. Like I said, yes, probably not even half a mile away from our property lines, probably less.
Yeah, well, that is very close.
If I could chime in real quick on that strategy too, I think if I recall for like EAB, for example, to save ash trees, they recommend if EAB is detected within 15 miles, I think it is, of your ash trees to begin treatment. So, obviously HWA is not spreading near as quickly as EAB is. So, but, I think I think it's reasonable maybe to consider treating trees if you've got, like Tim mentioned, known infestations within maybe a quarter mile. If you're using imidacloprid, that treatment's effective for 5 to 7 years within the tree. And it's, if it's that close, it's probably likely within that 5 to 7 years, it's going to show up in your area. So, and a lot of times, I'm surely it's on the trees that we survey, even though it's not on the low branches that we can reach. So if it's nearby, it could certainly be on those trees, even if you're not detecting it on those low branches. So.
I'll ask a question. Just regarding stem injection, you folks with the state who have just been doing hundreds and hundreds of trees, were you also finding more difficulty like midsummer, late summer, or different aspects? Or I'd just like that process to go a little more smoothly for us here at Crane Hollow, because I think we'll be doing a lot of it next year or this year.
Michaela, if you're still on, do you want to provide any insights on that?
Yeah, I honestly, it just really depended on the day. I didn't really notice, certain conditions that made it go a lot easier. Some days you were just going to be waiting a long time for that tree to take up the chemical, and other days it would go a lot more quickly. I think, it might be more individual, like weather dependent a little bit, but we were doing trunk injections into October and not having much trouble with it, but just again, depending on the day, some days were faster than others.
Did you guys notice anything with time of day being better, like early mornings or?
Not really, honestly.
Thanks.
Was there any indication or feeling that the soil moisture levels played into that at all? Or is it all over across the board?
I felt at Crane Hollow, I was having more success in moisture soil, like closer to the streams. But then we were just trying to treat some big ones, so we didn't go over per acre limit as well, like in a little drier, more xeric conditions. And yeah, I was just having trouble with those getting really anything. Like it was just, I was really trying to just jam it into the tree, and it was a long process.
Joe, I can comment on this as well, because we injected like 600 trees this past year. I have noticed actually some time of year differences. We usually omit doing treatments in the middle of summer when it's really hot. We try to stick to that late spring. But we also found going into fall, because being far enough north, we get really cool mornings. The trees just would not uptake the chemical at all. You would sit for like an hour and a half on one tree. And then as the day progressed and we got closer to noon and the sun came out or something, peeked out from behind the clouds, we noticed uptake would then move a little bit more rapidly. The other thing is depending on how deep you drill your hole, if you're noticing that you're struggling, and Tom and I had talked about this over the summer, is that, you know, you can drill your hole a touch deeper as long as you don't exceed a certain, what was it, Tom? Like, don't go beyond 2 inches. Try to stay in that 1 to 1.5 range.
Yeah, I would agree with that. 1 to 1.5 inch below the bark. Yeah.
Okay, and I might have been going up. I was trying to be a little not go too deep into the tree, so because I would, but it will uptake through the xylem, like still like pretty deep into the tree though.
Tom told me the first inch is the most effective area, but you know, it depending like how much chem you're putting in. Like we are using an optimized rate that Tom suggested to us, which is like 1 mil per dBH inch instead of what was like 1.5 mils or 2 mils. So that drastically changes how fast it takes it up. If you're going to do the higher rate, then it's going to take a lot longer. So you might need that little bit deeper cavity, or else you're going to really be sitting there a long time. And are you doing syringe method or?
Yeah, we're syringing it. I think Martin developed it, correct?
Yep.
And and it got easier too because we were trying to finish off some Imajet 5 and we moved to 10, which of course made life easier. And I just had a question too with that. Is it Zytec you guys were using? Is it basically the same thing as Imajet, like chemical composition wise?
Yep, same exact active ingredient, 10% imidacloprid. Yep.
Thank you guys so much.
Yeah, not a problem.
All right, I'll jump in here if that's okay. In thinking about a lot of the different presentations this morning, many of you guys used volunteers for various surveys. And Dave Atkins, if you're still on and listening through ODA, I know you guys work in partnership quite a bit with OSU Extension and heavily promote the use of Glidden. And so I'm sort of curious about, standardizing a little bit more when we do volunteer outreach and trainings. There's some internal needs that we all want from our data collectors. But as far as, collectively reporting or having a single source location, is there benefit to that? Because Glidden is used, for spotted lanternfly and Atlantis, but I don't think I've ever really heard anybody talk about Glidden for any hemlock pests. And maybe Dave Apsley would be a good one to kind of talk about that too. Sort of curious.
Yeah, that it's definitely a big problem because everyone's got their own preferred collection system, and it is hard. And even APHIS is changing their what they're collecting on. They're not using collector anymore, so they're going to be moving to a new collections device. In fact, they're having training on it today, right now. So, but that is a problem, but we've been able to work through it. We've got some good GIS people here that have been able to collect the information off the Glen and stuff and incorporate it into our numbers and so forth. But yeah, it is definitely a big problem, but because everyone has their own preference on what system they like. And so I don't know how to really correct it unless everyone can agree on one.
Yeah, we on our HWA webpage, we have a link. It links to EdMaps, which is the system that the Glidden uses. So I would recommend to the public or volunteers if they see suspect, or actually you can even report a negative survey on there. At least the page we had set up on our website, you can do that. I'm not sure about Glidden, but I think Glidden's probably the best option because we do push the use of that for other invasives. So I feel like it would make the most sense for the public or volunteers to report.
And that's very important, Tom, that people do report negatives even. So we know people have been in that area and looking, and we don't have to go back and repeat surveys in those areas.
And I agree. Tom and I worked on that for a long time. But I think, the EdMaps is what we've used on agency type properties, so we didn't overwhelm Glidden, but Glidden is the obvious place, I think, for private lands folks to report, and it goes pretty much in the same place.
So quick follow-up then, who among ODA, Division of Forestry, or OSU mine that data? How do you guys then go in and create reports from Glidden to look for those public reports from volunteers?
I know that myself and Kathy Smith, I'm not sure who else, get those HWA reports whenever they come in.
Yeah, we work through with Amy Stone to help us get that drawn down from the Glen.
So I'm really excited that we have Dr. David Mozell and Dr. Elizabeth McCarty here to round out our meeting. So I'll introduce Dave here first, who's going to give an update on just sort of an overview of biological control of HWA with Laricobius beetles and the Leucotraxus silverflies. So Dr. David Mozell is the regional entomologist for Region 9 of the U.S. Forest Service. So that's the Northeast and Midwest states. He is enrolled in the Muskogee Creek Nation of Oklahoma in the Fox Clan and is of Austrian descent. Dave was raised in Western Massachusetts and has a bachelor's in forestry from the University of Massachusetts, a master's in forest entomology from the University of Washington, and a PhD in forest entomology from Virginia Tech. Before joining the Forest Service in 2020, he managed forest insects, pathogens, and invasive plant issues on the Menominee Indian Reservation for 9 years. So Dave, thanks so much for being here to talk with us today, and I'll let you take it away.
Hey, thanks, Tom. Yeah, I'm also another hat I wear is the regional coordinator for the Hemlock Woolly Adelgid National Initiative for the Eastern region. In Region 8, or the Southern region, Rusty Ray is my counterpart. So I'm going to talk a little bit about classical biological control, and thanks, Tom, for the invitation. I'm going to turn my video off to conserve some bandwidth, but I'll come back later with that. Okie doke, let's go forward. A little bit about state and private forest health protection if you are not familiar with this group. Oh, I just lost my screen. Hold on a sec, Tom. Okay, am I back?
Yep, looks good.
Okay, so Forest Health Protection is a total of 35 employees in the Eastern region, in 4 offices. So I'm one of the regional office staff in Milwaukee. I'm actually in Shawano, Wisconsin, which is a bit north of Green Bay on the land of the Menominee. And the field offices are located in St. Paul, and for you guys in Ohio, your contacts are in Morgantown, and then New England is Durham, New Hampshire. So there's 12 entomologists, and Ivich Fraser out of Morgantown covers Ohio. You probably all know Ivich, and I think she's on the call today, and she's out of Ann Arbor, Michigan. Forest Health Protection includes pathologists, foresters, biologists, and remote sensing specialists. So we are here and available. Give us a call anytime, especially reach out to your field offices for any kind of technical or financial support, technical guidance, and And really, if you need help formulating responses to native pest outbreaks or biological invasions. Okay, so Eastern Hemlock and HWA. Here's the most updated map, including, and we've seen this today already, a bunch of new counties in Ohio and also New York, and then it's really spreading up in Nova Scotia as well. And the South has pretty much been infested for a long time. So the area of HWA infested hemlocks is scattered in this brown over 160 million acres and continuing to expand into some really heavy hemlock areas, as you can see with this FIA data in northern New England. Insecticides are super effective and a great strategy for treating high priority stands. So that's part of the IPM program. But on this regional scale, if we're thinking big, biological control with a mix of IPM activities just mentioned, to not put all our eggs in one basket of biological control, there's numerous strategies that can be employed. And I'll go into that in a little bit more detail right now. So the HWA initiative, we just wrote a new 5-year plan, and I don't expect you to read it on the right there. It's just a 2-pager document that highlights, our vision and mission and high-level priorities we feel all our partners should focus on in the next 5 years. And its purpose is to provide focus and direction. And it's not meant to detail your local tactical decisions, that we heard a lot about this morning, which is great, all great stuff, or stifle any creativity, but just to kind of have some pathways and to organize things across the region. So it never hurts to go into what our vision and mission is. So the vision is just conservation of hemlock, including Carolina hemlock in the South. The mission of the initiative is to accelerate development and implementation of effective management options through financial support, technical guidance, and interagency coordination. That's important as we're in this, HWA is affecting many agencies and landowners from private to federal and everywhere in between. So the 4 core strategies, this is the second page of that 5-year strategic plan, or the first page actually, the second page was the one we just saw, highlights the 4 core strategies on top. And we've heard about all these things already today, and this is nothing new. Biological control, obviously a great tactic for an invasive species, resistance breeding and restoration, seed collection, all that stuff, leading towards viable hemlock replacement options in the worst case scenario of all the trees dying. Insecticide treatments at high value sites and periodic retreatment. And also something that hasn't got as much attention is the possibilities for silvicultural treatments to restore and maintain hemlock health, depending on landowner objectives. So silviculture, there's data coming out and papers published on releasing trees from some competition, getting increased light or increasing resources, growing space. It helps, may not be in and of itself a solution, but helps with these other 4 core strategies. If you put them all together, especially biocontrol, especially with climate, if you're in cold areas, you have winter kill and you have the trees as vigorous as possible, you may see some increased survivorship and maintenance of tree vigor. So those are the 4 legs of the stool. And of course, all this is supported by the other great work that y'all are doing that I heard about today is the survey and monitoring, tracking HWA spread, and biological control establishment and hemlock health, information transfer. So there's various meetings, there's the HWA website, which needs some updating, and then there's some biocontrol and developing insecticide database of where you all have been treating these high conservation value areas that we would like to put into one map so we can know what's going on at a national level. But that's a work in progress. Research, always all these 4 legs of the stool need applied research and technology development. And lastly, the joint effort that I mentioned in this shared stewardship type all lands effort that we're trying to do. Okay, so that's just a little bit about the initiative. Let's get into one of the strategies. I don't have enough time to talk about, or the expertise to talk about the resistance breeding, but I wish to spend more time on silviculture, but maybe another time. Let's talk about the biological control. And it never it never hurts to go over what our goals and objectives are. And these things are really simple. The goal is ambitious. It's the long-term suppression of hemlock woolly adelgid below damaging population levels to prevent tree mortality and allow the regeneration of forests in damaged areas, for example, in the South or some of the mid-Atlantic states that have been infested for a long time. Biocontrol objectives are, again, really simple. The release of safe and hopefully effective biocontrol agents, safe for non-target impacts, for example, monitoring their establishment and the impacts on HWA populations, but also ultimately just the tree health. It's really what we're after here, to keep the trees alive and growing. And then the last objective, when the opportunity presents itself, is a redistribution of biocontrol agents that become established. We'll get into that in just a second. So the biocontrol strategy is the release of natural enemies of HWA from either the Pacific Northwest or Japan at this time. There was some foreign exploration for predators in China, but none of those species are really under much work anymore at this time, although we may go back to it at some point in the future if need be. So we need predators of both generations. This is very simple. There's no one silver bullet, unfortunately. You never know in the beginning. There was hope that maybe one of these species would just do enough predation on HWA to save all the trees in the region, but it doesn't look like that's the case. But the cystins generation, you have these two Laricobius species that Tom mentioned. They're doing a great job of establishing and spreading. So we feel like we have this cystins generation covered with a good, effective predator. We don't need much more work here. We do need work here in this Progrediens generation, which is, the spring-summer generation. Many species have been released since 1995. We've been doing this for a long time. Sasagi Scymnus sugi is from Japan. Scymnus cyneonodulus is from China. Scymnus coniferum is from Pacific Northwest, and the new kid on the block is these silverfly species, which I'll get into a little bit more detail. So there's one species, Pinnaperta, that feeds on the spring generation, and then Argenticollis appears to feed on both generations, with this fly having 2 generations per year. So let's go over Sasagi Scymnus sugi. It's really not something we're going to focus on much more in the future. There's been releases across, from northern Georgia to Maine, in area, almost area-wide, a nice distribution, starting in the mid 90s, like I said, and mostly stopping around 2018. These beetles are not available for free. You can purchase them, but we're not really recommending it. It's been recovered, and sometimes in high numbers, especially in the northern regions of the region. But the numbers just don't seem to increase like you'd like to see with a successful biocontrol agent. So they're persisting. They must be having some impact. I mean, they feed on HWA, so they're out there. They're probably a contributor, but not much more manipulation that could be done with this species. As you can see, this is a heat map of the recoveries. Oh, I'm sorry. I should explain. The blue squares are releases, and the purple circles are recoveries. So something to watch out for if you're if you're monitoring biocontrol agents, something to keep your eye out on. If you find them, that's great. And the heat map of recovery shows that there's really no massive numbers of these species being recovered. So it's a game of numbers. Without enough predators out there, you're not going to have the impact on the HWA, which has a massive biological potential for population growth. Scymnus cyneonodulus was released, and none were recovered ever that I'm aware of. There may be something in the south, I've heard, but I don't have that data at this time. So again, perhaps a failure. Perhaps they just show up in another 5 or 10 years. We're not sure. But it's currently not available for release. Scimnus coniferum, same deal. This was something from the Pacific Northwest. Feeds on the Progredians, was released at a few sites, and there hasn't been any recoveries. So that's a little background of kind of where we've been, and where we're going in the future is we want to focus on the Laricobius species and the silverflies. Tom had some great pictures of the fly here, and this is a picture of Laricobius nigrinus. We want to have some really nice guidelines for you all to refer to in terms of how to work with these species, how to release, how to monitor, and how to redistribute. So that's going to be a big project in the next year for me to work on with several other co-authors. It's really amazing how much work there is to do in biocontrol yet, even after all this time. But we're always improving and trying to make this as effective a program as possible. So some guidelines will be coming soon. There are some available, but they're in need of revision. So we want to support this Laricobius redistribution program because it's established at many sites, and the more we could spread them around, the better to increase dispersal and impact. And we're going to also increase the Leucotoraxis effort and integrated pest management demonstrations. I'll go into that in a second. So let's just talk a little bit about the Laricobius species biology. So we have two species, nigrinus from the Pacific Northwest and Osaka-hensis from Japan. Otherwise, they're pretty much very similar. They have one generation per year. They're dormant in the summer, just like HWA, so they're very synchronized phenologically. They're host-specific, which is always good. The adults feed on assistans nymphs, and the larvae feed on the Progredians' eggs. So as you can see in this diagram from Jeremiah Foley at Virginia Tech, he made this nice. The beetles are in the soil in the summer. They emerge in October, climb up trees or fly to trees and start feeding on the adelgids through the fall. And depending on your climate, it can be active or partially active during the coldest months. And they start laying eggs as soon as the cystins start laying eggs of the spring-summer HWA generation. The larvae go through 4 instars, drop to the soil again to pupate, and then the cycle repeats itself. So there's one generation per year. Leucotoraxis, the silverfly species, are relatively new, and we're learning stuff every day about their biology, but they have been determined safe and permitted for release. But we always want to know more, as much as we can, about the biology of the species. So there's work continuing. Both species are native to the Pacific Northwest. They also native to the eastern U.S., but in the Pacific Northwest, they're different genetically and appear to be adapted to hemlock woolly adelgid, where hemlock woolly adelgid is also native. So on the East Coast, these species, the biotype on the East Coast is adapted to various adelgids on pines. So we think by introducing this biotype is a worthwhile effort. Leucopus pinniperta has one generation per year. They oviposit directly on hemlock woolly adelgid. They overwinter as pupa on twigs right near the adelgids. Again, they're host specific. The adults don't feed, but the larvae or the maggots feed on that other generation that we need better control on. So the Progredians and cystins eggs. We're hoping that this is the 1-2 punch, a perfect combination of Pacific Northwest predators that really can affect sustainable long-term biocontrol of HWA. And there's been work in the Pacific Northwest that has shown that Laricobius and Leucopus are the main players and do regulate populations of HWA. The question now remains is, are they adaptable enough to the East Coast, and can that be replicated? Because it's just a different environment, different natural enemies, a whole different landscape. So That's what we're hoping for. It's good news that they're effective in the Pacific Northwest. The question still remains can we replicate that in the East? They also have, there's parasitoids that are common that lay eggs, or parasitic wasps that lay eggs on species of flies. So you have to be very careful about collecting them because we don't want to introduce any of their natural enemies. Leucopus argenticollis is very similar, except I mentioned already there's 2 generations per year, and they feed on, the maggots feed on both of them. So this is just my interpretation of the life cycle of the silver flies. I think the silver fly experts have not put a life cycle diagram together, perhaps because there's still questions remaining. So this is from the literature, and I think the best state of our knowledge of what's going on here. So Leucotraxis argenticollis. Here, let's look at this species. The pupa overwinter. In the spring, the adults fly and lay eggs, and they have 3 larval stages, pupate, and all feeding on the adelgids. And then pupa emerge, and there's another flight in this Progridians generation, spring, summer. They go through those same developmental stages again, and then they overwinter. So I hope that makes sense. 2 generations per year. Leucotraxis pinniperta has just 1 generation per year, so they have a little bit of a longer overwintering period. The adults fly later, and lay eggs on the Progridians and do most of their impact on that generation. So this is a nice complement to the Laricobius species. We have predation for argenticollis, both generations of adelgids. Laricobius really hitting this winter and spring generation hard, and then we got pinniperta feeding on the Progridians generation. So really bracketing the life cycle of the HWA with predators, and that's what it's going to take to control something that has asexual reproduction and can just reach massive numbers really fast. So how do you go about collecting and rearing Laricobius species? This is what's done in the Pacific Northwest and also in the eastern U.S. So in the Pacific Northwest, you have the anywhere where you can find adelgids on western hemlock, there's a potential to collect Laricobius beetles. So we've scouted all over Oregon, Washington, and northern Idaho, and collected beetles, quite a few, and we're able to release those in the east after a little bit of quality control screening. And it's really pretty simple process. No one's ever looked in Southeast Alaska. That would be an interesting place to go, but perhaps not really necessary. What's a big distinction is there's this population of beetles on the west side of the Cascades, and also in the northern Rocky Mountains. So there may be some differences in cold tolerance between these 2 populations. So we collect from both and release beetles from the northern Rockies into areas like New England or other cold high elevation regions. And these coastal beetles are just doing great in the mid-Atlantic and southern states. Before we realized that adult collecting in the Pacific Northwest or establishment in the East happened, all the work was done in lab rearing, very labor intensive, costing up to $20–25 per beetle, which requires, maintaining the beetles in the laboratory for their entire life cycle, feeding them in environmental chambers, in plastic containers, as they oviposit on adelgids, and then moving that foliage to these funnels. And then the larvae develop in these funnels and drop into these mason jars, and then the larvae have to be transferred to soil containers, and then you have to wait, many months for them to emerge in the fall again. So very, very labor intensive, but has been very important for getting the numbers of beetles out there in the environment that we have today. So Leucotraxis collecting is very simple. There's no rearing of its complete life cycle in the laboratory because it just hasn't been figured out how to do that yet. What is done is foliage is collected in the Pacific Northwest, just like just the same places Laricobius is collected, but you collect foliage with heavy adelgid populations, you ship them to quarantine laboratories, and then the pupae close and the adults fly, and then the adults are collected in these cages. This is actually a Sasagi skimnus cage, but rearing cage, but it's similar. The adult flies are collected and then shipped to releasers. So this has to be done in quarantine facilities. I'm not sure if I mentioned that, and those are at Cornell, the University of North Dakota, and then Virginia Tech is going to be coming online. They have federal quarantine facilities to make sure. With raw foliage coming in from the Pacific Northwest, there could be all kinds of insects on there and pathogens or things on western hemlock that we don't want to introduce. Also, the parasitoids of the flies, we have to be careful. So all has to be done in a very controlled environment. So it's not a DIY project by any means. So the release strategies. The Laricobius, again, I'm going to wait till we have our guidelines. There's so much information to cover: how to pick a release site, how to make a release, pick a release tree, and enter information into our national release database. I'll just keep it really simple here today. So stay tuned for that document. We hope to have it out in a year that'll really outline the step-by-step process on how to work with these two species. But basically, Laricobius species are released in a short window in the fall. This is all based on their life cycle, because this is when they emerge from the soil. So this is when we release them. You can collect them out west, you can collect them out east, or you can get them from the lab. Same in the spring, there's also a short window where you can release, because the beetles oviposit on the HWA eggs right here. So if you release them at this time, they'll be able to, potentially avoid the winter environment and start to lay their eggs and have their larvae develop right here. Okay, so two windows for release. The primary one is in the fall, because that's when it's easier to collect the adults, and the rearing facility, they don't want to hold them in the laboratory for a whole winter. It's just expensive. But let's not forget about the spring opportunity to release Laricobius. This is what it looks like when you get them in the mail or hand delivered. Is the beetles are shipped on, Excelsior, which is safe to release. We don't want to be shipping hemlock foliage around that may have elongate hemlock scale on it. So we use this kind of inert material. You drape that Excelsior on the tree and let the beetles disperse on their own, or sometimes fly off. Okay, Leucotoraxis species. So again, just based on their life cycle, it's really important to get the timing right. So here, the silver arrows are Leucotoraxis argenticollis, and again, they have 2 generations per year, so you can make a release in the early spring, overlapping with Laricobius, so you got the 1-2 punch, and then you could make another release in the Pregridians generation, so the 2nd generation argenticollis. Leucotoraxis pinniperta can only be released around this time in the spring and early summer. Okay, I hope that makes sense, and it's very, very simple. So let's look at the status of where's Laricobius at. Okay, the first release was made in 2003, and that was a graduate student at Virginia Tech, Ashley Lamb. She made an egg release in Southwest Virginia on the Jefferson National Forest, and that actually established. Took a couple years for populations to build up, and then I went out there and recovered a single adult beetle, and it was really exciting. And then I launched into my PhD research, which was releasing from northern Georgia to Maine, and that was up to 2007. And a lot of those sites had a beetle establishment too. So going on all the way fast forward to 2021, and again, the blue squares are all the releases, including releases in Ohio, like Tom mentioned, and recoveries all over the place. This is a voluntary database, so I think these recoveries are conservative. Not everyone enters the data, but as you can see here, it's pretty inspiring to see that the beetles are adapting to this eastern climate and building numbers significantly. I mentioned that Northern Cascades, the Northern Rocky Mountain region, biotype of nigrinus that may be better adapted to cold areas. So we made those releases starting in 2007 once we discovered that the beetles are out there in Idaho and Montana and Northeast Washington State. So we made releases a lot up in Maine and Massachusetts, in New York, and some of those have established as well. I guess I didn't include the circles where they've been recovered, but so we're also hopeful on that for colder regions. And if you lump them all together, the nigrinus recoveries in this heat map, we're starting to see significant numbers building up, especially in this Delaware Water Gap area. Beetles have been dispersed up to 30 miles out of some of these hot areas. Western Maryland is also a very good area, and they don't have all their data quite entered in the database yet, but I think this would be very hot, just like New Jersey and Pennsylvania. Also in North Carolina, the beetles are starting to appear to be everywhere, also in Southwest Virginia, like really spreading across the landscape on their own from initial release sites, to the point where, okay, so I mentioned that biological control, the Laricobius rearing facility at Virginia Tech, so they're really spending their efforts trying to rear Laricobius osakensis right now because it's harder to get to Japan, and there hasn't been as many releases yet. They collect foliage all across Southwest Virginia to feed the Laricobius osakensis, and they're bringing in Laricobius nigrinus just from the environment, sometimes not even near release sites as they're rearing. So as they're rearing in those funnels, osakensis, they're getting nigrinus bycatch. It's just an example of how this thing's really starting to take off, and it's taken a long time, but we may be on the verge of something really, really great happening. Tom, you still got a good audio on me? I should probably check periodically.
Yeah, still looking and sounding great.
Okay. I think I got, how much time do I have left?
You've still got half hour.
Oh, okay.
Including questions.
Okay, plenty of time, and I hope to leave a lot of time for questions. So this is just a map. I don't know if y'all know where Blacksburg, Virginia is, which is right here. This is where that rearing lab is. And beetles have been released in these blue squares in this area of Southwest Virginia, and recoveries. A graduate student did a great study. He said, let's go just look in the town of Blacksburg on suburban trees or urban trees, and also in the town of Radford down here, not anywhere near a release site. And they're finding beetles have been recovered all throughout these little towns on ornamental trees, also in natural forests, up to 30 miles away. This is Hollins College in the city of Roanoke. Really quite impressive. I'm not sure if we have a good handle on how far this beetle is dispersed, and that's a good sign of progress towards the ultimate goal of suppressing HWA below damaging levels is dispersal and population increase, and we're seeing both of those things happening. Okay, osakensis is that other Laricobius species from Japan. It's been released from Maine to Georgia again. Not as many because this one wasn't discovered until later, and this first release wasn't made until 2012. Recoveries have been made throughout the areas that it was released, including Maine, which doesn't show, they haven't entered their data in, but Maine has some new recoveries just this year, so that's exciting. There's been recoveries in Ohio, same with nigrinus. And so again, although it occupies the same niche as nigrinus, it's just more options in the tool bag. It may be more effective. The beetles are bigger, are actually, I don't have a picture of them, I forgot to put one in there. They're slightly bigger. They may be more voracious predators. That's been observed in the laboratory. They may be more cold tolerant or less cold tolerant. We don't know. So we're releasing both, and we'll see who wins. But that's osakensis has a little catching up to do. Recoveries have been made but not huge numbers yet, except for in Pennsylvania. I'll talk about that in a second. I think there was a recovery in Ohio. I'm not sure why it's not showing up on this map, but it shows up on this map. Anyways, let's keep going forward. The silver flies. The first release was actually done quite a few years ago, but there really wasn't a good understanding on exactly how to, the timing and the life cycles. Even though it was studied and determined to be safe in terms of host range testing and approved by APHIS for release. But some of these early releases maybe aren't that reliable. But recently, New York State has really decided that they're going to focus on leukotoraxis, and they've done a ton of releases. Just last year, we did some big releases in Pennsylvania and 2 sites in Ohio, as Tom mentioned. And then there's some releases in the South. So there hasn't been any recoveries yet, unfortunately. We're hoping the flies are adaptable. They're a little less, they're flies. They're not as tough as beetles are. So we're hoping that they're able to make this transition, getting shipped overnight by FedEx and reared in a laboratory and then released and be able to reproduce in the field. So it's a big ask, and we're hoping to just get the ball rolling here. More news soon. It will be breaking news if someone recovers a Western biotype leukotoraxis in the field. So we're eagerly awaiting that to happen. So let's get to the bottom line. Are these things effective yet? I mean, could you sell them and say you're guaranteed 100% control of your hemlock trees? No, we're not even nearly there yet. We know Laricobius species have significant impacts. Numerous papers have been published about the level of predation and predator-prey ratios in Virginia and elsewhere. And they're showing, impacts have been documented. So they're having an impact on that generation that they feed on, but it doesn't appear to be enough on its own. So we need additional predators, and I mentioned that already, and that's where we're hanging our hat on leukotoraxis. If leukotoraxis doesn't work, we can go back to China, where there's a beetle called Scymnus camptodromus that was almost ready for release, but the colony was lost. The colony died. It became really difficult to get back to China to recollect those beetles. So that's something we're kind of holding on to, and maybe in the next 5-year operational period, if leukotoraxis doesn't establish, we have that in the pipeline. Okay, so leukotoraxis, we really have no idea of what its impacts are going to be in the east and out west. Yes, there's been indication that it's part of the predator complex that regulates HWA, but it hasn't even established in the east. So that's the first hurdle, and then we can start looking at effectiveness on HWA and tree health. So I mentioned biocontrol guidelines. So please stay tuned. This is going to take a while to write, and we need a lot of input from people in the field, from various states and agencies to really line out how to release, how to monitor, and how to redistribute beetles. We do have, I did mention, we do have drafts that are available on the HWA website, but we need to revise them because there's nothing in the current guidelines on silverflies or monitoring impact or redistribution. So a great example is the document that's been published by the EAB biocontrol program, where they have similar guidelines. So redistribution, let's review the work that occurred this fall. So like I mentioned, Laricobius, nigrinus, and ossakensis have established many sites and are increasing in significant numbers. So in Maryland, Biff Thompson with Maryland Department of Agriculture and his crew here, and I believe Tom was a part of this group on another day, they went to Rocky Gap and they were able to collect over 5,000 adult Laricobius nigrinus in a relatively small park. And that's an incredible result. The true population size, who knows? There's probably hundreds of thousands or maybe millions of beetles out in the environment. They're only using beat sheets on the bottom of the trees, so you can imagine that there's a significant impact on HWA there. So they collected all these beetles and they relocated them to 4 different states and a total of 9 new release sites. So this is the natural phase of a biocontrol program moving forward and hopefully eventually to the successful outcome of saving trees. That's what it comes down to. So this should be very exciting. It's been done before, collecting and moving beetles around, but these HWA program managers, Biff and others, and Tim and Tom have really stepped up the game this year. Also, Scott Salem and at Virginia Tech, him and his crews went out and collected over 1,000 adults in Virginia and relocated them to Northern Virginia at 2 sites. Western North Carolina, been doing this work for a long time. A private entomologist has his own company. He collects and redistributes them in North Carolina. So he's collected over 155,000 beetles since 2008 and spraying them across the landscape. At some point, you could stop doing this once they're already established across the landscape, and they disperse on their own as well. But it does help to speed the population growth and dispersal if we can help them along. So we hope to do this more in the future, grow this redistribution program, maybe even hire a 2-person crew to dedicate, the entire week to do this work and move the project forward as fast as possible. Osakensis. Kind of surprising that Tim Toman in Pennsylvania at a release site was able to collect 1,684 adults at one site with a crew of several people. And these were relocated to Maryland and Pennsylvania for a total of 4 new release sites based on 1 successful establishment. So Osakensis is a few years behind nigrinus, but we expect that the results to be the same. Okay, I mentioned we don't want to become target fixated on biological control alone. There are other IPM strategies, of course, that I mentioned earlier, just briefly, the 4 legs of the stool. And if biological control just doesn't work, we need backup plans. And one of the best backup plans is the resistance breeding and restoration planting. There's other strategies that are available, silvicultural treatments, and obviously insecticide treatments of high conservation value areas. So I thought I would just show what an IPM program might look like in, from my perspective anyways. It really depends on what you can do. Depends on the land you're working on. So some people might not actually be interested in biocontrol. There's always concerns about non-target impacts. There's some people that don't have logging in their mission, or national park, for example. We've heard a little bit about there's concerns and legitimate concerns about insecticides, and restoration planning has a lot of work to start bringing trees that are tested and proven to be resistant or tolerant of HWA, and those are ready to be planted. So various combinations of this possible, but let's just do a hypothetical example of what could be done at a state park in Massachusetts. And this is where I grew up. We used to play pond hockey here on Bray Lake and have keg parties up on this hill over here. And now the hemlock woolly dove has been there for a while, and it's really sad to see a lot of decline in the trees in this area. And the Mass DCR has been doing work here. I'm not all up to date on what they've been doing, but this is just a hypothetical example of how would you implement an HWA IPM program here. So this is a parking lot, and you got the lake here. So maybe around this area, you'd be selecting trees for insecticide treatments. Also over here, is the mass state record biggest hemlock in the state. So I know this has been treated. So that may be something you want to treat, right? Like, who wants to take a chance on biocontrol working on the state record tree? So you got your insecticide treatments. Logging is probably a tough proposition in Massachusetts, but just pretend this is hypothetical. Maybe in this interior forest area, you do some silviculture, do some single tree selection, maybe with some special hemlock marking guidelines to give the hemlock tree some growing space, some vigor, some light to tolerate HWA damage. Also, in Massachusetts, we get the climate effects of cold winters. So cold winters coupled with increased vigor of the trees may be effective at prolonging the life of the trees. Okay, what about biocontrol? So, I mean, I want to release biocontrol agents right on insecticide-treated trees, but you could do that right near them. What about a cluster of a release on a cluster of heavily infested trees right here? You know, and then given a few years, these beetles will disperse throughout this whole area. Even in the insecticide-treated trees, I mean, they're not going to feed on an insecticide-treated tree if there's no adelgids on it anymore. But you may have untreated trees in that area, and that's been shown. And there's publications on how to integrate biocontrol and insecticide treatments. And lastly, restoration planning. So this is just hypothetical again. Say there's a bunch of hemlock mortality down in this area. This is where the infestation started. Maybe there's opportunities for planting bulletproof trees or something that came out of the breeding program eventually down here. So, you know, I've worked for the Menominee tribe on managing 235,000 acres. So I've been there. I know it all comes down to your local forest and your park, and you know, really making your own plans. And I know the states all have their own conservation plans. So that's the fun stuff, and I miss that. So, anyways, hopefully that gives you some ideas, and you're thinking about your properties and what you can implement and how to do it. And that's all up to you. The tactical and the strategic. The tactical decisions and timing and what you can implement is all, and creativity, and there's probably lots of room for creativity and innovation here. Is all up to you all. So that's my last slide, and I just want to acknowledge all the HWA program managers. You know, we just had a big national meeting on HWA biocontrol, the biocontrol working group, and there's over 130 people there. So unfortunately, you can't list them all, but people have been doing this work for a long time and implementing the tactics on the ground. So appreciate all the people. And lastly, there's a steering committee that guides the HWA initiative, and that's led by not just Forest Service people, but it's chaired by the FHP director of Region 9. And we have David Arnold from Tennessee, and he represents the National Association of State Foresters. Kimberly Rice from Maryland represents the National Plant Board. And we got Tom Macy himself representing the Northeast Midwest State Foresters Alliance. And Keith from APHIS, and then some Forest Service entomologists from the Southern region, the Washington office, and the two research stations, the Northern and the Southern research station. So this steering committee guides a coordinating committee, and we kind of work to think big picture strategic planning. So hopefully that all made sense to you all, and you got a picture of options that are out there. And that's all I had. I hope I got you thinking, and there's some questions.
That was great, Dave. Thanks so much for that rundown on biocontrol. And yeah, I think that note you closed on is really important too. I mean, it's going to take more than just one of those stools to really reach our goal of hemlock conservation across a large scale. So that was awesome. Do any folks in the meeting have questions for Dave on biocontrol or anything else? Yep, I see Rob has his hand raised.
Yeah, I had a question. I was just curious, and others may be able to answer this better. Just with biocontrol, we got stuck with the same thing when EAB happened, and I think it went through Department of Ag, but I mean, we just went through kind of the four legs, and you know, if someone decides to use insecticides or silvicultural practices, it doesn't really affect anyone else off-site. But with biocontrol, you know, the rest of us have to live with whatever's, when we pull something from Japan and introduce it, and it hasn't been well tested, and it has, you know, alternate hosts. I mean, I'm all for biocontrol, but I feel like there's too little buy-in from you know, other agencies and organizations that are going to be seriously affected, and just wondered if there was a way that could be. I'm just curious about comments on that. We kind of felt like we had to go with EAB biocontrol, or didn't have to, but even though we didn't want to, we felt like it wasn't probably the best thing to do because of some alternative wood boring host species that were going to be affected. We felt like it was already being done. It was going to be there anyway in our parks. We might as well, you know, bring it now and try to save a little bit of ash if it's useful. You know, we just kind of had to concede. And so that seems a little different than a lot of the other practices. And I feel like probably a lot of us feel like we're just kind of afterthoughts or in the process, but didn't know if anybody had any comments on that.
Yeah, I mean, that's a good point, Rob. I mean, there's once something's released and established, there's no really taking it back. You know, it's persistent. Nothing more persistent than a established biocontrol agent that's spreading. You know, so ultimately, as far as you know, I'm new to the Forest Service, about 2 years. But as far as I know, all these species were you know, are ultimately approved by APHIS through categorical exclusions, or EAs. So that their scoping process, I'm not totally sure, but in my old position, I remember getting letters from APHIS to the tribe saying, "Hey, we're releasing, you know, EAB parasitoids in Wisconsin. So do you guys have any comments on that?" And we never really did. But yeah, I feel it, and it's definitely, if there's non-target impacts, that's a bummer. We're fairly confident that these are safe. These predators are safe, and they've gone through all the host range testing that was required. And
well, we do know that they are going to affect the pine adelgid, or very likely could, because you know the one biotype of this silverfly, I think, also hosted in Eastern North America on the pine adelgid. So bringing the other silverflies in from other places, or like, you know, could wipe those out. We don't know.
Yeah, no, totally a legitimate concern. There's been really good DNA evidence by Nathan Havel at in Northern Research Station that has shown that there's different subspecies that are focusing on these not subspecies, these biotypes are staying focused, at least in the Pacific Northwest, on their host of preference. So it's another line of evidence that matches that laboratory host range testing, that it may not make the switch, but it may. I mean, those are a legitimate question, and that's you have to monitor. There'll be, once the silverfly is established, if they establish, there's monitoring plans in place to investigate hybridization and host switching. So that should be part of a post-release monitoring program, and that probably will be in a guideline. So let's monitor what's happening on pine bark adelgids. Nothing's really come. There's been no red flags with Layer cobius on also on pine bark adelgid. Lab studies have suggested that it's not a viable host. It doesn't really match the life cycle of that. But the flies are, different. So, it's hard to go back after you release something. But best you could do is to monitor the situation, and hopefully there's no problems. A risk versus reward, cost benefit analysis is the classic example of that type of decision making.
So I'd like to jump in here, Rob, because I'm a little confused by the premise of your question. Because one of the things you just mentioned was when you decide as a land manager to treat your tree with insecticide or do other silviculture, that it only affects your land, whereas a biocontrol release affects other people. And I would argue the fact that, a lack of treating trees and then potentially that infestation serving as a source for insects that are readily spread via hitchhikers through birds is just the same kind of issue or concern. So I didn't feel like I had to release those biocontrols for EAB. We at Cleveland Metro Parks actually never did, but I think fundamentally I think I disagree with the argument that your decisions on your land from a silviculture or insecticide treatment are only affecting the infestation on your land. I'm sorry, I just couldn't help myself, Rob. For those of you that don't know us in the Northeast region of Ohio, we have often spirited conversations.
Yeah, and I will follow up with you offline, unless somebody wants to hear my reason.
I was muted. I see Matthew Smith with his hand raised.
Yeah, so my question is, so let's just say that you started somewhere you did an aggressive insecticide treatment, for your HWA, and you decide to potentially maybe start integrating or switching to a biological control. What do you have to consider? Because, is that insecticide is probably going to infect whatever biological control you release. So what are the considerations on if you would like to do a biological control someday? What do you have to consider beforehand when you're treating with insecticides?
Tom, you want me to take that one? Or is that for me? Oh, okay.
Yeah.
You know what? I'll put a link. Bud Mayfield at a Southern Research Station, along with a bunch of co-authors, wrote a nice guideline document. Oh, there it is. Exactly how to do that. So, I mean, there's been shown to actually not be a significant impact if the timing is right. I mean, you don't want to treat the same season as a release on that, on that tree, but maybe so there's spatial considerations and temporal considerations, but they're not incompatible. And thanks, Tom, for putting that there.
Yep, I agree. I think just what Dave said, yeah, that this document, this publication would be really good to check out. But yeah, I mean, you don't want to release beetles right onto a tree that's been chemically treated, but I think in close proximity, it's okay to do if all the trees aren't treated, and that I think that is something that we're looking at and starting to consider where we have infested areas that, maybe it's more than just one little isolated infested pocket and it's becoming more or less generally infested. Maybe we switch up that strategy to only treating a subset of the hemlocks there to keep them alive and healthy, meanwhile releasing nearby on our untreated trees that would hopefully sustain the biocontrol.
I guess to follow up then, if you would like to look into biological controls, what considerations Tom, does the Division of Forestry or whoever would permit that? What do you what are the considerations you have to make before you will, approve or of a biological control in an area?
Yeah, I mean, there's some guidelines on this, and Dave can chime in too. But I mean, in my mind, the most important thing is having enough HWA there, to support a population of the biocontrols. I think in a lot of places right now where we're finding HWA in Ohio, there's not a ton of adelgid there, and I worry that if we put biocontrols there, they won't take hold, because you get a cold winter, it could almost completely knock out that HWA population. But I think you need a pretty substantial HWA population, and ideally hemlock's still healthy, and there's good, low hemlock foliage that you can easily release and potentially collect from to redistribute beetles in the future, or silverflies. But yeah, we're always on the lookout for populations of HWA that could potentially support a biocontrol release. And so we're starting to find some new areas that I think would be good places to release them. So that's, what meetings like this will help us with, figuring out where there's good sites for doing releases.
If I'm not mistaken, sorry, it's Rachel again. I think that document that you just put in the chat goes over those biocontrol necessities for the population numbers, which is, something that's super important. And I think that, like you said, it's one of those things where we're aware that we're releasing something different into the environment, but at the same time, we have a lot of failures, and then in the end, on most of these, we hope that they just predate each other out of existence, so that when HWA goes, then the biocontrol goes out of population as well. And that always makes me feel a little bit better about bringing in the different species in order to take care of the situation.
Yeah, thanks for that, Rachel. Cool. Oh, I guess I missed a question from Connie there. Are there biocontrols for elongate hemlock scale, or is there any effort to find one? I am aware of one that I'm pretty sure was identified and reared for some time, and Dave, maybe you know more on this, at New Jersey Beneficial Insect Lab, Mark Mayer. It doesn't have a common name, but it's Cybocephalus niponicus, which I think has become fairly widely established in the Mid-Atlantic. I'm not sure if there's a lot of data or studies to show how effective it is, but I think possibly as we continue to explore more of these established HWA biocontrol populations in the East, as we encounter Cybocephalus, which is the predator of elongate hemlock scale, maybe there'll be potential to collect and redistribute it as well.
Yep, we're probably out of time, but I don't know much about Cybocephalus myself, but I think it's been collected in Michigan, where they also have elongate hemlock scale. So that's promising. Definitely a player. Lastly, Roy Van Drijch and one of his grad students did spend some time in Japan and identified through various methods several parasitoids of elongate hemlock scale that appear to be host specific. So that would have to go through the whole slew of host range testing and quarantine evaluation. But funding kind of ran out, and then there was retirements, and people moved on. So that's something we're looking at getting reviving. If I get trying to throw that out there to researchers who wants to pick this one up, it wouldn't take a ton of money. Could be super effective. There's a lot of precedence for successful scale insect biocontrol projects in all different kinds of ecosystems. So that one could be a slam dunk, especially if it's get host specific parasitoids and safe and so on and so forth. So that would be maybe 10 years down the road, but there's potential there too. That's a good question. Thanks.
Awesome. Thanks, Dave. All right. Thanks. Yeah, so welcome back, everybody, for our final presentation today. Really glad that we have Dr. Elizabeth McCarty with us. Elizabeth is a forest health outreach specialist with the Warnell School of Forestry and Natural Resources at the University of Georgia. She received her PhD in entomology from the University of Tennessee in 2016. Her areas of specialization include forest entomology, aquatic entomology, insecticide use in trees, and environmental risk of insecticides. She has active outreach relationships with University of Georgia Extension, Georgia Forestry Commission, many forestry companies, the National Park Service, U.S. Forest Service, and forestry and natural resource agencies in many states. And her current research includes integrated pest management for pine tip moth control in young pine stands and non-target insecticide risks to pollinators, ground-dwelling arthropods, and aquatic insects in forested systems. So Elizabeth, thank you again, and feel free to take it away.
All right, thank you so much. Please chime in if you can't hear me at any point. So thank you for having me here today. This is going to be a talk about trade-offs. We would love to have a perfect situation where we don't have to get into the nitty-gritty of these trade-off decisions. But everything has a positive and a negative, every single management decision. There's always going to be a risk and a benefit and trade-off. So hopefully through this talk, I'll share some data and help as we think through those things. Let's see. All right, so I'll very briefly mention HWA impact on forest health, the insecticide treatment options, again, very briefly, because I'm preaching to the choir here. And then I'll go through some environmental risks in soil arthropods, water quality, canopy arthropods, and pollinators, and some of these will kind of wander in and out of each other just because of how the studies were designed. Some of this will be my own research, some of it will be research of others. So this is not just me, this has been work from numerous different scientists, and I want to go ahead and just acknowledge that. So our resource is eastern hemlock, but eastern hemlock is not our goal. It is a very important tree species. We also have Carolina hemlock, but most insecticide treatments are on eastern hemlock, and that's where I focus. It's got this distinctive ecological niche. It's slow growing, long lived, shade tolerant. It really has an ecological role that cannot be replaced, and many different plant and animal species depend on the presence of eastern hemlock in our forest. So the goal is not to save a tree. The goal is to preserve an entire forest ecosystem. And so that's important to keep in mind. Everything is connected. Things don't stay where we put them. And that includes the effects of HWA. And we've got this forest health threat, HWA, and y'all already know the situation. We've had widespread hemlock mortality. And what we're left with is a post-HWA forest. So I started working with Great Smoky Mountains National Park in 2011. I went to the University of Tennessee and did a project that was park funded. So essentially, my entire HWA experience has been in forests that are impacted. And so if you are on the leading edge of the infestation, you're getting to see an HWA forest or hemlock forest that I haven't gotten to see very much unless I've gone to visit it. And so what we have after hemlock mortality is these open canopy gaps, a rapid vegetation response. Especially in the south, we get increased rhododendron density. And so if you're not familiar with rhododendron, it's called rhodohells for a reason. They're awful to get through. Things can't grow under them. And there's all these altered aspects of the system with vegetation, with light, with nutrients, with water. And so that is part of the trade-off when we start to talk about insecticide use. And so I'll share some information. Note that sometimes what people think about insecticide use and insecticide effects, it is not in keeping with what is true of insecticide use and insecticide effects. And so we can talk about that a little bit more in the Q&A time, but I think maybe most of y'all already are living with having to deal with stakeholder concerns. So we want a healthy forest, and again, it's not about a tree, it's about an entire ecosystem. And so insecticides are used for conservation. I'll mention dinotefuran and imidacloprid because those are most commonly used. Dinotefuran moves up to the canopy quickly and is effective for about a year or two, whereas imidacloprid moves to the canopy more slowly and is effective to 5 or 7 or more years. Dinotefuran is great for a short-term reduction of HWA, whereas imidacloprid does provide that longer term control. Dinotefuran is more expensive. It doesn't last as long. So that is the tool that's best used for heavy infestations, whereas imidacloprid is best for light to moderate infestations. And so we have these two very complementary tools. They're both from the same insecticide class. The non-target impact data has been focused mostly on imidacloprid because that is the tool that is most commonly used. But they're both from the same insecticide class, so there may be some differences. But let's stick with what we know about imidacloprid and know that there should be some similarities. Let's walk through this environmental risk scenario together. So let's see. I can get the laser pointer. Here we go. So we'll start with pouring imidacloprid on the soil because that's how it's most commonly applied. We'll move up into the canopy with expected uptake into the hemlock tree to kill fluid-feeding insects. There could potentially be some impacts to non-target canopy arthropods. And I'll pose that it would be pretty silly to think that we're going to treat with a broad spectrum insecticide to kill canopy feeding insects and expect to have no non-target impacts. That's just not a reasonable expectation. There's going to be a trade-off in this situation. And then the question is how much, what amount of impact is acceptable? And as a scientist, I feel very strongly that I provide the data and appropriate interpretation of the data, but the resource managers are the ones that have to make the decision on, are these trade-offs reasonable? And so that's a decision that is in your area. I can't make those decisions for your management program, right? So we have potential impacts to the canopy. We could possibly have impacts to soil arthropods, movement through soil and uptake into non-target plants, including floral tissues, which could have the potential to have impacts on pollinators. I'm not really concerned about pollinators for hemlock canopies because hemlocks are wind dispersed. So that pollinator route would come from movement into non-target plants. And this picture shows these giant trillium plants that we could walk under. Really what we're talking about is small plants that are growing close to the base of the hemlock tree, and potentially some larger shrubs, but all things that are growing very close. We don't expect impacts that are outside of that canopy line. And then imidacloprid could move through the soil with potential impacts to surface water quality, including aquatic insects. And when we talk about risk in these systems, the organisms that are most sensitive to imidacloprid and in pesticides in general are generally aquatic insects. They are very, very sensitive. So when we think about pesticide movement in the environment, and this is true for imidacloprid, hemlocks, or any other situation, first, is the insecticide present in the environment? Okay, we've applied it. Is that residue still there? How far has it moved from the application site? How high is the concentration? Just because we find it under the tree where we poured the pesticide, we would expect that. But are we finding it in high concentrations moving away from the tree? And is that a problem? Could this concentration that I found be a problem for an organism? We can detect concentrations at very, very low levels, but just because insecticide residue has been detected doesn't necessarily mean it's a problem. And so then we have to put those concentrations in context with the biological system, and that starts with lab tests. But I'll caution people, an effect that we get in a petri dish is not always the same effect that we'll get in the lab, in a biological system. It's a good starting point, but those results should not be taken too far. So then the next question is, does the concentration that I found cause a problem to organisms in natural settings? And that's where we have those bioassessment data, which is incredibly valuable, because if we see an effect in a petri dish and we don't see an effect in the field, then that's pretty powerful comparison there. And then what are the trade-offs for natural resources management? So do we have an effect? How much? How far away? And then what are our trade-offs? So let's talk a little bit about imidacloprid residues in the environment. These are all specific to hemlock systems. Some of this is my data, most of it is, a lot of it's data from others. And so this is just a way to put everything in one picture. First, the limit of detection. So how far down can I detect a concentration? And for most of the time, it's going to be about 1 parts per billion. That's how far down we can detect. In water, because aquatic organisms, especially aquatic insects, are more sensitive, typically those are down, and I usually go to about 20 or 15 parts per trillion. So a much lower concentration that I want to be able to detect in water. So what does it look like? What I and others have found in water is concentrations that range from the limit of detection all the way up to 1 part per billion. All right, and I use a kind of a gradient here because most of the time the concentrations I detect are less than 100 parts per trillion. So most of them are in this lower range. What happens when people tend to cite this kind of data, especially if they're telling a story, is they will pull the highest concentration observed and then phrase their whole argument based on the highest concentration, which is really inappropriate. We don't do science by outliers. We need to know what's going to happen most of the time to make a decision that is most meaningful for the situations that we encounter. Secondly, soil, and this is from a study by Nope et al., where they used lysimeters and looked at the concentration of water that is in the spaces between the soil. All right, so around 1 to 100 parts per billion. Hemlock foliage, 1 part per billion up to about 1 part per million. So that's the range we typically see. That's how much we need to kill adelgids. And not to get too far into the weeds, but when we have imidacloprid in foliage, we also have an insecticidal metabolite that works with it to give us that longer term hemlock control, HWA control. From a pilot study in rhododendron that a colleague and I did, our pilot data, we get about up to 1 part per million in foliage, and then a little bit in the flowers. So foliage and flowers, we get lower concentrations in rhododendron foliage than we do in the flowers, and that's typically what we do see in flowers compared to foliage. And then lastly, on a study that I'll present the data, looking at concentrations in bulk soil. So the data was collected in a different way than the previous soil, but a giant range. And these are collected within 2 feet of the base of a hemlock tree, a big range of what we find, but most data points are lower down in here. In non-target plant foliage, and these are growing within 6 feet of the hemlock tree, and then in flowers. Again, most of the concentrations lower down, but we do have those outliers that are higher. Again, those are outliers, so we don't tell the story by outliers. So this is just to give an idea of, yes, we do find imidacloprid moving in the environment. These are about the concentrations that we get in the environment. And then we have to pair that with biological data to see what that actually means. I mean, we could take chemical data and tell a big story with it and run around with our hair on fire, but that doesn't necessarily mean that that intensity of an effect is happening in the environment. That just may be an emotional effect. And so we have to pair all this with relevant biological data. And then the question is, how does this change over time? How does this change moving away from the tree, and what does it mean in these systems? So every management decision, like I said, has consequences. We're looking at, this is me in grad school, doing biocontrol recovery. We've got native insects on the tree, HWA, insects that pupate in the soil, canopy arthropods. And so every management decision we make will have consequences for many aspects of the biological decision. And that includes the decision to do nothing, and we can't discount that. Here is a hemlock canopy that has not been treated with insecticide, and we see what's left. And so the concern of maybe some minor impacts of pesticides to a hemlock canopy, when we make that in the trade-off of what happens to that hemlock canopy if we don't treat, and this is the easiest, this is an easy one. Some of the other risk scenarios are not as easy to talk through, but we can see that when the hemlock tree is gone, the native insects that feed on hemlock trees no longer have a habitat. And so we have to decide, are those trade-offs reasonable? Are they good trade-offs? Are they bad trade-offs? And then know that the consideration that we make as scientists, as land managers, may not be the same thought path that the general public takes. And so when considering these type things, we've seen this. We can take a couple of different perspectives. Some people choose to take an inflammatory perspective, and we see this in a lot of news headlines that are very simplistic, and no amount of environmental risk is ever acceptable. And this is where we get a lot of that science by outliers. And then we may have a dismissive perspective that totally disregards non-target issues. The focus is only on this management goal, and this objective has to be met, and any amount of trade-off, any amount of non-target issue is just, it's something that they don't regard. And neither of these perspectives, are very helpful. And so we end up in the middle where we want to land with a balanced perspective, looking at the entirety of the data, that information embedded in the biological system, because facts are stupid. A fact out of context has no meaning at all. We have to look at the entirety of the data in context of the biological system, balancing trade-offs and considering different perspectives. So let's try to land somewhere in here. So if we could put this on a balance and have the consequences of hemlock loss and the consequences of hemlock management, and if we could have this perfect equation where we could balance that all out, that would be great, but we don't have that. And note that there's some things that could be a consequence of hemlock loss that could also have consequences from hemlock management choices. And so this is a very messy area, and we're going to do our best knowing that we may never have a perfect answer. And then when I talk about this, I like to always share this quote: for every complex problem, there is an answer that is clear, simple, and wrong. Let that sit with you for a minute. So we're talking through complicated things, and I know this is a lot of intro to it. I'll kind of fly through some of the information, but the perspective, the thought, how we think about these things, how we consider them is incredibly important when we talk about non-target impacts. So again, I'll wander through 4 different areas, and let's see how we get through the data part of it. First, aquatic macroinvertebrates. This is largely from my PhD project, and so this is a map of Great Smoky Mountains National Park and an area showing where we collected water samples and aquatic macroinvertebrate samples. I'm doing another study that's funded by the Forest Service, so we're out in the Smokies in North Carolina and in North Georgia doing more water sampling, more aquatic insect work. My lab's full of bugs to ID, so this work is being continued to continue to look at risks. And so yes, sometimes we do find imidacloprid in surface waters. Typically concentrations are less than 100 parts per trillion. Occasionally we'll get a spike of a higher concentration, especially when it's associated with a rain event. So to lay this out, the red box indicates a treatment area. So this is where hemlock trees and the riparian corridors have been treated with imidacloprid by basal drench. An upstream sample would represent a control. A downstream sample represents where we would expect to see an effect if we have one. So this is the treatment area. And then the baseline represents Great Smoky Mountains did have some species list collected before HWA was ever in the park, and so that provides a baseline of the taxa that were in those streams before imidacloprid was ever used in the park. And so the comparison is made between all downstream locations and all upstream locations, and this was taken from 9 different streams in Great Smoky Mountains National Park. The focus is on mayflies, stoneflies, and caddisflies. Those are very sensitive aquatic organisms, and so if there is a problem, these are the taxa that we would expect to be most likely to see it with. And so this is comparison of the downstream areas to control locations. So we've got 6 different community metrics. Abundance is the total number of insects. Richness is the total number of species collected. And then we have dominance, Shannon diversity, and evenness. Those are just different ways to look at diversity in the community. And lastly, mean tolerance value. This is a water quality metric that's used with a lot of regulatory programs where different species are given different tolerances. So the lower the number, the more sensitive the insect, and the higher the number, the more tolerance. That gives us an indication of the types of insects we're finding in these streams. So again, downstream is where we expect to see an effect. And we can look at these numbers and see that going across, they're pretty similar. And in fact, no significant difference was detected between downstream and upstream and downstream and baseline from all of these different metrics. So we're not seeing an effect in these sensitive aquatic insect communities from the imidacloprid treatments done in Great Smoky Mountains National Park. These are again all done with, according to the label, using basal drench or soil drench treatments and not exceeding the maximum rate per acre. Is that to say there could never be an effect? No. We could always do a study with less soil with less organic material, or somebody could exceed a label rate and do something stupid. But we do know that in this system, following the label, we didn't detect an effect. And on top of that, I also looked at different other aspects of the community like functional feeding groups and life habits. So I took a pretty close look. I just don't want to overwhelm you all with numbers. The take home point was looking at multiple aspects of the community. We didn't see an effect. Another study was done at University of Georgia. This was not my study. It was done with the Forest Service Southern Research Station. And imidacloprid treatments were applied in the riparian areas of 4 streams. They also had a control stream, and they sampled water and aquatic insects for 2 years after application. Only 1 sample was positive. It was right around 1 part per billion, but their detection limit, as time goes on, we get better at analytical techniques, so their detection level was higher than we're using now. And that was only collected 2 years after treatment. Again, extensive aquatic insect data collection and analysis, no negative effects were seen. They didn't see a problem, and the aquatic insect metrics indicated excellent water quality at all of these streams. I do have another study in process, and I'm not at the point where I can comment on that just because we're early in on the identifications. Next, canopy arthropods. Okay, if we see an effect, this would be one of the more likely places to see an effect. This study was done by Carla Dilling at the University of Tennessee. I worked with the lab with her. This was an excellent, very, very comprehensive study. Canopy arthropods were sampled by 5 different sampling methods for 1 to 2 years after treatment. There were some negative effects on abundance, some negative effects on richness, but not catastrophic. So looking at soil drench, let me see, I took a picture on my cell phone so I could reference this quickly in case I couldn't access it on my screen. Soil drench, horticultural oil, soil injections, and tree injections. So numerous application methods. And in general, with some of the metrics, all application methods, including horticultural oil, had significant effects. A little bit lower abundance. The effects were a little bit marked with soil drench, but again, it wasn't a complete wipeout. For instance, on controls, on average, 180 taxa were collected from hemlock trees, where it was 155 with soil drench. So some reductions in the species collected, but it wasn't a total wipeout. And that's important to know to qualify that. There were 33 species that were directly affected. So this is where we see the effect, not on everything, but moths, and a lot of these pupate in the soil, and then bark lice, which are pictured on the bottom left-hand side, and these are detritivores that are feeding on the tree. A second study was done by Falcon and DeWald. This was a bird study, so they looked at canopy-dwelling arthropods as prey for birds, and they did not see a difference in order-level assessments. They did see some effect in relative abundance on treated sites. So we do get effects in canopy arthropods. That's not surprising. Again, not a wipeout, but some reductions. And then another study was done by Kung et al. This is further north, where they looked at trees 3 years after treatment and 9 years after treatment. There was no negative effect of imidacloprid treatments 3 years after treatment, so a little bit of a longer term window. And then 9 years after treatment, there was actually higher species richness on treated trees. In this case, the untreated trees were still alive but had declining canopies, and so that effect was seen due to canopy decline in untreated trees. This section will be a combo of pollinator risks and soil arthropods, just because there's some overlap. This is mostly highlighting a study I've just completed and I'm in the process of wrapping up. So this was a study done in North Georgia. The purpose of the study was to look at non-target impacts for persistent trillium. This is an endangered plant that grows in 6 isolated subpopulations in North Georgia and South Carolina. It is only found in mixed hemlock and hardwood groves. And so trillium needs habitat created by hemlocks, pollination, and ant-mediated seed dispersal. So there's 2 types of insect associates with persistent trillium. And this came to my attention. There were some Forest Service land managers in the area that were like, hey, we have to conserve this plant. We have to keep the hemlocks there, but we're really concerned about this trade-off with non-target impacts. And so they contacted me and we put together a study to help answer this question. And then it also has applications outside of this area because we're missing that part of the puzzle about potential impacts to pollinators and at that time movement into non-target plants. So here's the persistent trillium issue. We have habitat. It's threatened by HWA. Pollination and seed dispersal, again, we get these cascading environmental effects of losing hemlock, and we don't know the effects of it, so they could potentially be impacted by loss in HWA. Insecticide could impact the insect associates. And then let's add to it Asian needle ant, which has moved into this site and is in the process of wiping out our seed dispersing ants. And so this is an incredibly messy system, but as land managers, you end up dealing with these messy situations quite often. Often it's not just one problem. Which problem do we want to pick to deal with? So the project objectives were to evaluate the risk to persistent trillium pollinators, and that risk is evaluated by looking at the concentration of insecticide moving into the flowers. Pollinators are hard to assess in these systems. They move in and out of areas, and so it's just much more difficult to do that assessment, but we can look at concentration and get an idea of risk. And then evaluate the risks to those seed dispersing ants. There were 2 sites that were located near the Georgia-South Carolina border and 15 study trees at each site. They were divided into soil drench, soil injection, and bark spray. So when I treated these, it gave us also the opportunity of people had started doing bark spray maybe like 7 or 8, 9 years ago, and they started saying, well, this is more environmentally friendly because it doesn't, you're not treating the soil. But nobody had actually assessed that. And so is bark spray actually, and I don't like to use the term environmentally friendly because it carries a lot of connotations, but does it produce less of a risk? And so that gave opportunity to look at that issue as well. So for a picture, our risk, again, we've got these small plants that are occurring adjacent to the hemlock tree. These are all herbaceous plants less than 1 foot tall. And I've got this picture of a ton of flowering plants right at the base of the tree. Sometimes, like if you're in a hemlock silverbell system in Great Smoky Mountains National Park, this is what the herbaceous layer looks like, and it is full of herbaceous plants. On the slopes that I'm working on in North Georgia, not so much. So the degree of risk also depends on what your herbaceous plant layer looks like. Treatments, 5 trees in drench, injection, and spray, and that's repeated at 2 sites. And the samples were collected were soil, foliage, and floral samples. The goal was to collect 2 samples of each type under each tree. I'll just go ahead and say because of low plant density, we on average only collected 1 floral sample under the base of each tree within our specified distance. And so that does say a lot about risk if you're not getting a lot of flowering plants growing under the tree. And so that really depends on your situation. What does your forest floor cover look like? And that has a lot to do with the risk. And then leaf litter samples to collect ants. The insecticide residues were done by mass spec by Anthony Legalante at Villanova University. And then a timeline. So we started in March 2018, and these are our controls before treatments were applied. I treated the trees right after collecting the samples, and then 3 weeks later, collected the first samples. And so the floral samples, the first set, those are 3 weeks after application. So this would be a highest risk scenario. 4 months later, in August, we collected again soil, foliage, and leaf litter. And then everything was collected in April and March. We didn't collect floral samples pre-treatment just because there were not enough flowers there to do that. 1 year later, a year and a half later, we got a break in 2020 because we went into lockdown right before I was supposed to go out. And so then we have 3 years post-assessment and 3.5 years. And so, you know, the hemlock treatments are effective for 5 or more years. And so this carries us about halfway through the treatment time. Let's think about these risks, all right? When we collect a sample under a hemlock tree, that concentration under the hemlock tree does not translate to the entire forest. And it doesn't necessarily translate to the entire plot of land. So we've got a polygon here, and then we've got these islands of risk that are occurring under the hemlock trees. And so this is about a 6-foot circle around the base of each hemlock tree. So the entire polygon is not affected, rather these little islands of risk around the base of the tree. So it's important to keep that in mind. Soil was collected within 24 inches of the tree, so that's right where the applications are being made. Foliage, we had to expand out the foliage where we could collect because we didn't have that many plants growing within 24 inches of the tree. So that's within about 62 inches, and then flowers within 74 inches. We had to expand that out a little bit more. And so that's the area of risk. All samples were collected on the downslope side, so that's where the pesticide would migrate as it's moving away from the tree. Persistent trillium was the goal. However, there was not enough persistent trillium to collect, and so we ended up collecting from the herbaceous plant layer in general, with almost half of our samples coming from persistent trillium. And so now walking into the data to set this up, this is soil concentrations of imidacloprid. So this is the topic of the table. We've got the imidacloprid concentrations in soil and parts per billion. And then we'll lay it out at the control, 3 weeks, 4 months, on to 3.5 years. I'll first state that there was no effect of treatment type. So drench, injection, and bark spray until we got to 3 years after treatment. So no effect at all. Same concentrations observed in the soil for bark spray treatments as the other application methods. When we get to 3.5 years, there is some change, but right now I'm just focusing on the time aspect of it because that's more applicable for land managers. And if you have any questions afterwards, you're welcome to ask. And so at the beginning, okay, we essentially have no imidacloprid, tiny bit from treatments 8 years before. 3 weeks after, absolutely, those concentrations dip up and they remain high, statistically similar for about 1.5 years. Although we do have a big dip, those concentrations are very variable, so we don't have a statistically significant difference for about 1.5 years. We get to 3 years later, it drops significantly, and then 3.5 years, it's similar to those control levels. So look on the y-axis, we've got concentrations in thousands of parts per billion. Are these concentrations levels that could hurt, you know, perhaps an ant moving through the soil? It could, but these are done by bulk soil, and so not necessarily all of that is biologically available to an organism. The moisture level of soil has a lot to do with the toxicity of soil. So a soil sample with similar concentrations but different water content can have different toxicity to insects. And then I also collected samples during August, 10 feet downslope from the trunk. So whereas within 2 feet, it is 4,500 parts per billion, 10 feet away, it's 19. A year and a half, we were like at 1,000, 1,500 parts per billion, down to 21 parts per billion, 10 feet from the trunk, and all the way down to 0.3 parts per billion 3.5 years later. So those concentrations that we find right at the base of the tree are not the same concentrations we'll find moving away from the tree. And so as concentrations reduce, risk is reduced. So as we move away from the tree, the risk is lower. Second, we have those ant samples. So these were collected in leaf litter samples right at the base of the tree, so right where we have poured the insecticide. And we've got the number of species and the different treatment methods, no effect of treatment method on the ant community. The number of species, again, are richness, and over time, we had some issues 1 year post-treatment, so we had to exclude those samples, and those were just site collection issues, but no effect of time on ant richness and ant abundance, or the number of ants collected. No effect of the different treatment methods on abundance, and no effect of the time on abundance. So we do get those soil residues, and at the same location we collected ants, and we're not seeing an effect. We've also collected bycatch from those leaf litter samples, and we'll be looking at other taxa. That data has not been analyzed yet, but we're going to continue to take a deeper look in that leaf litter dwelling community where those organisms are in and out of that soil. And then this is a different study I'll mention at the same time since I'm talking about soil arthropods. This was by Nope et al. Done at Coweta Hydrological Lab, where they looked at treated and untreated trees, and there was some imidacloprid movement in the soil vertically, but not very far, like down into the soil column, but not very far horizontally. Again, their concentrations, I shared this at the very beginning in that first figure, were much lower than the ones I observed with bulk soil, and they used lysimeters to collect that data. So they do see movement vertically, but limited horizontally. And then their soil arthropods had no population difference between treated and untreated trees. There was a weak negative correlation between imidacloprid concentrations and insect populations at one site, and that was where it was collected 20 to 50 centimeters deep. I'm hesitant to really dive too far into this because it was a very weak correlation, and it was only collected at one site. And so I don't want to go too far out on that one since it's a single site collection, and we try to look at data that's collected from multiple sites. Right, next back to the Trillium study, looking at imidacloprid concentrations in the foliage. Again, this is foliage collected within about 5 feet of a hemlock tree. And so we've got the control. There's a small amount, and one of these sites did have previous applications 8 years before, 3 weeks, 4 months. And then note that we see a trend where earlier in the year, it's a little lower, and then it increases non-significantly through the growing season. And that's because as those plants continue to sit and accumulate water and take in water, they're accumulating imidacloprid during the growing season. And then the plants die back, resprout in the spring, begin to accumulate over the course of the season. And so they'll tend to have that cyclical pattern with insecticide residues. And so concentrations increase and stay up for a couple of years and then go back down at 3 years after treatment. Again, could this concentration potentially, kill a beetle that's feeding on a tree? Some of it could. We're looking at a limited area, so there is some risk, and then we have to take that risk and put it in perspective to the entire forest stand. And then I like to share this picture. It is when I was applying a bark spray onto a tree. I used a low flow sprayer and did very, very careful applications, and we did get some bounce back onto the foliage. And so that is a concern with bark sprays. And when we did that that did happen with persistent Trillium, and that's where I got my highest residue levels. All right, and then lastly, floral. This is where we get the pollinator risk. And so again, 3 weeks after treatment. So this is going to be our highest risk. And yes, we did have high concentrations in those flowers that are growing right adjacent to hemlock trees. The flowers bloom, they die off, and then 1 year later, we're at about 25 parts per billion, and then 3 years later, we're at less than 1 part per billion. So through the life of that hemlock treatment, those concentrations don't stay very high. And if it's a sensitive area and an area that's concerned for pollinator risk, then this would be a great option for a fall treatment. And then we're not treating right before the flowers start to bloom. And then the EPA no effect concentration for honeybees is 25 parts per billion. And we have to be careful with these numbers. They're not a line in the sand, but they can be used to help us think through some risk. So this is the concentration at which no effect was seen in honeybees. And honeybees are not the pollinators of persistent trillium, but it's a good starting point. And so no effect is seen at 25 parts per billion. We know we're at that concentration 1 year after treatment. And so that tells us that we're at least in a range that's probably acceptable and that we're having incredibly low risk. So initially we do have risk, and that tapers off over time. So again, we've got these consequences of hemlock loss and consequences of hemlock management. And we've talked through some of the possible consequences of hemlock management. And so the question are these risks reasonable? There will never be no risk. We can never, say that nobody will ever apply a treatment irresponsibly. But this is something that I like to try to land on as we take the next steps, as you take this information home with you and think on it. Final thoughts, we do have some limited impacts to canopy arthropods. Again, not a total wipeout like could have happened and wouldn't be unreasonable for us to inspect, but some lowered abundance and richness with certain taxa. Some media in the environment, like the soil, does have higher residues than other, and that does decrease moving away from the tree. And so the questions asked are, what proportion of non-target organisms are affected by these residues? How much of a trade-off should be made? What are the consequences if we don't conserve hemlock in these areas? And then how do we wrap all this up with public perception, which is not always science-based, but something that we all have to deal with. So I'd like to thank, for the data that I collected, field assistance from Great Smoky Mountains National Park, Atlanta Botanical Garden, the University of Tennessee, and funding sources from my data, and then also acknowledge that a lot of this work was done by other people, and so this is not all my data, and to thank those people for their contributions to our knowledge base. And with that, I can stop sharing and be glad to take any questions.
Thanks so much, Elizabeth. That was really great. Just really awesome to hear about, research studies and publications that are out there on this, because that's what we need. We need good studies to inform these management decisions, as you discussed. So really awesome stuff to hear about. And I don't think I saw any questions during the presentation in the chat, but I see Rob has his hand up now. Go ahead, Rob.
Yes, I really enjoyed that. I think it was a very balanced view. And honestly, Tom, I really enjoyed the whole meeting today, so might as well say that now. My question or comment really is related to some of, I mentioned in my talk early in the day that we had documented some new species to science and some really small headwater streams that are associated with hemlock, which is part of why where our concern comes from and why we have been hesitating with using some of these neonics. But there's some other cases I know in Crane Hollow where they were talking about tiger spiketail and some of these headwater streams right in the middle of the hemlock area. They're using tablets there where there could be some runoff. I don't know if you did anything with tablets. I didn't hear offhand, but
I did sample a stream with tablets this summer, and we found a little bit. I mean, when you put a pesticide in the environment, we are getting some of it in the water, yes. But the question is, are we getting enough to cause an impact to aquatic insects? And
so I guess we have some scuds in some of our streams that, like I said, were new species to science. And, you know, you really opened it for a case by case in the beginning for managers. And I appreciated that because there are, I think, special circumstances in certain areas where you might have a really sensitive taxa. It might be the only place that it exists in the world. And, you know, just a small mishap could wipe something completely out or out of a decent chunk of its range for, you know, just like the hemlocks are in these isolated sort of patches in this part of the world. A lot of their host species or associates are as well. And some of them are new species or species that we haven't even found yet. So I just wanted to really emphasize sort of our approach in that area, and that some care is probably, I don't know if you tested that with any really rare species or anything where you were working with where you might have had just a species associated with a single stream reach or something like that. But we do have a few cases with those up here.
I will say that the EPA, the risk assessment, the lab risk assessment that has the most, I guess, where they've seen the response at the lowest concentration, I get that taxa in streams all the time where I'm getting imidacloprid at, you know, these lower concentrations. The thing about assessing rare species is that it's kind of hard to do unless you're doing a study specifically for that one species. But I do appreciate your point that there is absolutely room for this by case basis. And if you as a land manager feel like protecting a species of invertebrate in a stream is your biggest priority, then that's your biggest priority. And you definitely have the freedom to do that. There's no one perfect answer for doing this. We do have a range of things you shouldn't do. I mean, there are some things that are bad management decisions, but there are lots of good management decisions.
Yep, thanks for that. I also see Ivich had her hand raised. Go ahead, Ivich.
Hi, great talk. I was wondering if you could, are there any studies on comparing effects to non-targets between systems that are healthy and those that are depleted? So environments that are, where you have a, you know, it's more of a management situation too, where, sorry, I'm losing my question here. Are there any studies comparing healthy environmental systems to poor or depleted systems and the effects of the non-targets for?
Oh, I've got an echo now. Can you turn your speaker off? Yeah, so that's an interesting question. I'll take it two ways. You know, we think about comparing imidacloprid to a perfect hemlock system, and that has been done in the past some, but that shouldn't be our control anymore. That perfect system is going away. And so that's just not the reality that we live in if we're getting HWA. But that's typically what's done. We take the perfect system or our healthy system, and we take the solution, the insecticide, and we compare those differences. I think a more appropriate comparison is comparing in systems where you're using imidacloprid as opposed to systems where hemlock has died. That is a more appropriate control at this point, because that's what you're going to get if you're not treating. That doesn't quite answer your question, but I think it does kind of get at what is our control. And we had to do that with the new aquatic insect study we're doing now. It's like, what's our control here? I wish it could be a perfect untouched hemlock system, but unfortunately we're losing that. Okay, I know there's another question, but I can't see from where I am who it is.
Yep, I think Derek had his hand up next. Derek Cooper.
Hi, Elizabeth. Thank you for your talk. Just had a quick question about, is there any explanation in the pathway for why basal bark treatment, soil drench, and injection showed no differences in the amount of chemical residue in the surrounding soil area?
All right, well, this is the first study that we've done on this, and I apologize for not being able to share my camera. It is messing up, and it makes me look like a ghost. So I did look at rainfall after we treated, and we had very little rainfall, because my first question is, well, maybe, we had a big rain after this, because that is one concern, is if bark spray gets washed off. I think my best pathway is that we apply the bark spray. It's not moving in as fast as we would hope, and it's sitting on the outside of the bark. And when it does rain, it gets washed out. But again, there was not a significant rain at this site for at least 2 weeks. And we can't, as land managers, say, well, I'm going to apply this treatment, but I have to make sure it doesn't rain for 2 weeks before I apply it. That's just not reality in a lot of our systems.
I think I'm not sure if it was Matthew or Joe. I'll go with Matthew for the next question. See your hand up.
It was Matt.
Okay, cool.
Thanks. And I don't know how quite to word this question, but in your talk you mentioned about the duff layer and how that would impact, obviously, the amount of insects and things that are living in that area and the effects that the insecticide would have on that. Has anybody had any consideration, like there's some areas like in northern Ohio and other areas of the country where there's the invasive earthworm that's coming in and almost completely removing the duff layer, and any correlation between, again, I don't know exactly how to word that, but the impacts of how that would be with some of our results? Or
I don't know about any studies on the invasive worms in hemlock systems and the duff layer. So that's a little bit out of my wheelhouse. And I think, unfortunately, there's not a lot we can do about the invasive worms. And so I try to focus on the things that we do have control over.
Yeah, and I guess more of my talk was, I thought you said something about how the amount of duff layer that was there, it kind of provided more of a habitat and less of a conveyance for the, I don't know if I heard correctly, of that insecticide to move through the system. The duff layer kind of helped buffer that?
No.
Did I hear incorrectly?
Really, no. So when you apply an imidacloprid treatment, what you want to do is remove that duff layer.
Right.
And then, you apply and you let it sink in and you put that duff layer back. You don't want a lot of that imidacloprid to be in the duff layer, because then it's not accessible to the roots.
Right, and that's what I guess that's where my question was formulating, where you don't have that duff layer and you can't put that back over top when you've done your soil drench, so it's kind of leaving that exposed, and I don't know.
Right, and you may have some risk right where you poured the pesticide. You are correct, but that is the area that we are going to have to accept some risk.
Right. I think Joe is up. I see your hand raised.
Yep. Yeah, great talk. I thought it was just interesting how it seems like the foliage of the herbaceous plants were increasing imidacloprid over the course of the summer. Does a similar thing happen with the hemlock needles, even from a year-to-year basis? Has anyone studied that, or is that, you might not know, but does anyone know?
Typically when we look at, there's a big difference there. These are herbaceous plants and they're dying back during the winter.
Yeah.
The difference is that in the tree, that's an evergreen tree, and those leaves act like a sink for the pesticide. So as water moves the imidacloprid up in the xylem, it accumulates in the leaf and keeps going to that one area. And then they're not falling off and then refoliating every year. So I don't expect that we would find that effect. We do kind of get some increases over time, and that is part of Carla Dilling/Coots' work. And then she did the first couple of years where she looked at how it increases over time, and it kind of steadied out. And then I picked up that work and looked at 4 to 7 years after treatment, and then we kind of see it start to taper, downhill a bit.
Her name was Carla Dilling?
So she published under Carla Dilling during her master's, and then Carla Coots during her PhD.
Thank you so much. And as far as like if somebody was treating for elongate scale, has anyone really done studies with that as well? Because I know a lot of times imidacloprid isn't used as much for elongate scale.
Imidacloprid is not great for scale insects in general. So it really, scales are funny things, and it depends on the scale insect as far as what insect, and this is just in general and ornamental work as well. Imidacloprid's not great for EHS. There has been a study on different products and the efficacy, and I have seen that data, but I don't know where it is in the process of publication. So I would be looking out for that in the next year or so, probably.
Thanks so much.
I don't see any more questions in the chat or anyone else with their hand raised. Does anyone have anything else for Elizabeth or for anyone at this point?
Yeah, well, I got you here, Tom again, and Paige and other people maybe, just for the stem injection. In your syringes, about what's your maximum you'll try and do for a single syringe? I'm wondering, like, because you could just drill a whole bunch of holes and only put like 2 milliliters in each. I've tried to do more than that, certainly, but I was wondering if that's some of my difficulty as well.
Yeah, I wonder, Michaela or Paige, do you want to weigh in on that?
We could, I mean, we fill the syringes because what we'll usually do is drill multiple holes and have our ports and maybe fill a syringe up all the way and then distribute that evenly among the holes. So, I mean, we would put in more than just a couple mils in one port, up to maybe like 5 or 6, I think, depending on the diameter, like the larger trees, and depending on how many good like root flares you can get ports in. And I'll also say, I mean, I believe according to the Zytec label, they recommend the number of ports to be half of the DBH. But we often do less than that. But if you're not, if the tree's not taking it up, you can always add more ports up to that half of the DBH if you need to.
Thank you.
Tom, I just put a link in the chat. It is to the risk document for HWA. So it does have most of the information that I presented, put in a way that's easy. So if you have citizens who are concerned, it's made for agencies and management groups to share with other people.
Excellent, thank you. I'm familiar. That's a great document. Thanks for sharing that. And then I see John has a question.
Yeah, I talked briefly about Worcester Memorial Park. We're about 20 or 25 miles northeast of Mohican State Park. Mohican has a lot of hemlocks, and it hasn't been mentioned in the talks today. And I wondered, you know, if they have been surveying, and I assume nothing has been found.
Yep, that's correct. I think we did mention it in our talk at the very beginning. We've been doing survey of Mohican every year and just wrapped that up a few weeks ago this year. And yep, nothing has been found there yet. So as far as we know, still free of HWA there.
Okay, that's good to hear. I missed the first few minutes of the talk, so I probably missed that.
No problem.
Thank you.
All right. Joe, did you have something else? I see the hand still raised. You might have just forgot to put it down. Okay, that must be the case.
I'm sorry. I didn't even know how to roast. I'm just
no problem.
I'm just stirring up the flow. I apologize.
You're fine.
I did want to just add, again, with Joe asking about the injection, I feel like, you know, it would be more of like a maximum of like 5-ish milliliters per port, but I would say I'd probably average about 3 milliliters per port in doing the chunk injections.
I was overdoing it then.
All right. Well, thanks so much, Elizabeth, for your presentation, and Dave again. That was really awesome stuff, and I think it was really helpful and enlightening for all of us, and myself included. So, thank you. All right. Well, that concludes our meeting. Again, if folks have, hemlock conservation concerns, questions, want assistance with workshops, we've got a lot of things that we'd like to do here in the near future, and we'll be reaching out to you folks to assist us with that, hopefully. So we'll keep in touch, and like I said at the beginning of the meeting, we're hoping to convene this group at least once a year. I know I've lapsed on that the last couple of years, but yeah, at a minimum of once a year, and if needed, we can get together more often than that. But so hopefully around this time next year, or a little sooner than that, I'll be in touch and we can get another meeting scheduled at that time. Maybe we can do it in person then. That'd be nice.